Good morning, and welcome to Friends of Cancer Research's 17th annual meeting. Thank you for everyone in the room for being here today. We really appreciate you coming out and joining us for a series of exciting discussions throughout the day. And I'd like to thank all of the people that are joining us virtually online as well. Each year, Friends of Cancer Research's annual meeting represents the culmination of several months of work where leading experts have come together and developed actionable steps to address specific challenges in oncology drug development. As we get started this morning, I want to thank all of the participants in these working groups and the team at Friends that has spearheaded the work to help lead them along the way. We're grateful for all of the time and expertise that you've been able to share with us the last several months. We'll hear from many of those experts today throughout the entirety of the meeting as we explore three different topics focused on approaches to streamlining clinical trial designs and data collection to support efficient regulatory processes and decision-making. But before we move into the sessions today, I'm excited that we're able to start with a terrific keynote this morning. And to get us started, before we move into that session, I'd like to introduce Friends of Cancer Research's chair and founder, Dr. Ellen Siegel.
Good morning, and thank you, Jeff. So we had quite a week last week, didn't we? But the one thing I will tell you about Friends of Cancer Research, and I believe everyone in this room, there are two things, many things we're committed to, and that's science and what's best for patients, and that's what we're going to do, and I believe that's what everybody is going to do because that is our responsibility. So we have an exciting keynote with Rob Califf. I've known Rob Califf for a very, very long time. They've given me tons of stuff to say about Rob, but you all know it, so I'll just say a little bit. I will say that I've known Rob for 40 years, and I've been tormenting him for all those years. So I always make his life complicated, but it's expected. From day one, Rob, taking the helm at the FDA, we began working together to transform clinical trials in this country to ensure they are better for all patients. We are incredibly lucky to have a commissioner who works tirelessly to advance evidence-based medicine and standardized electronic clinical trials. He is a true public health advocate. He cares deeply at a core level for every patient and science, and that we translate that not only to our prestigious centers, but to the community where patients really need the help. So, Rob, thank you. You're a tirelessly advocate for patients and for all of us. I promise to continue to torment you, though.
All right. Well, welcome everyone. My name is Ramsey Baghdadi, and I'm the co-founder of Provision Policy. I'm thrilled to be here with FDA Commissioner Robert Califf, and Dr. Califf is coming to the end of a really remarkable second term as commissioner. He's had to deal with everything from shortages on infant formula, saline, and certain cancer drugs to undertaking a massive reorganization, the Office of Regulatory Affairs and Human Foods program. The first full Alzheimer's drug approval and first accelerated approvals of gene therapies happened under his watch. At the same time, he's been commissioner for the full COVID vaccine approvals post the EUAs and the explosion of GLP-1 drugs and a decline in opioid overdose deaths. And Dr. Califf has also pushed major initiatives on real-world evidence, modernizing clinical trial infrastructure, and then how to best regulate artificial intelligence. And those are just a few of the things he's been involved in. And it's just an amazing number of accomplishments during his second stint as commissioner. So I've divided the discussion into several sections, and I thought we would start with the obvious one, which is the election. And I would love to get your reaction. This is an essay question. I would love to get your reaction to the election and how you think the change in administration will impact FDA's work.
Well, start out with a softball. Obviously, personally, I'm disappointed in the election, and it's not about me because I think it's pretty well known. I had not planned on coming back to FDA until I got a call asking me to come back. I was having a pretty good life in Alphabet Land, and so I came back, and I feel like from the FDA perspective, we are sort of got all engines on go right now. We've done a massive reorganization. We've got very significant plans. There's a meeting going on today that I just left, going over the hiring. We're hiring pretty well and not having trouble getting very talented people. And I think we just don't know what's going to happen now. So it's not correct of me to make any specific comments about exactly what will happen. But I do think it's pretty clear that the gist of this administration, from everything that's been said, is to change a lot of things. And how it gets changed depends on who gets appointed into key positions and how the various policies play out. And I also think how the broader, as FDA is concerned, how the broader regulated industries see things and play into this. So, in times of change and chaos, there's also opportunity. And I will just comment, there are a lot of things that I'll bet almost everyone in leadership positions in the health agencies would have liked to have changed but couldn't because of the sort of gridlock of various interests that made it difficult to change things. So I think this, obviously I'm biased, but I feel like the FDA is sort of at peak performance right now. And we'll just see what happens as the new team comes in.
Okay. I'm curious how you respond to the kinds of threats that have been made by people like RFK Jr. saying very specifically that he wants to clear out FDA groups that work in certain categories like nutrition and who knows what else. Because from the outside, we see that and we're like, oh my God, I can't believe he just said that. But you at the agency, how do you guys take that kind of threat? How do you respond to something like that? That's out there, it's going viral, it's being, it's all over the place.
In my role, it's best not to respond to those kinds of things because getting into a back and forth about hypotheticals is not productive for anyone. I will say, I'm not an expert on food, and my first term, Steve Ostroff had been the acting commissioner, and he did have food expertise, and he became the deputy commissioner when I came in and handled most of that. So this time around, though, it's been the majority of my time, for example, has been on food. And I don't think you have to be too smart to say we have a chronic disease crisis going on in this country that starts in childhood with terrible diets that are not getting better. And there is work that needs to be done. But, I still love Alistair Wood's old article in the New England Journal, Kick the FDA, America's Favorite Sport.
Yeah.
The things that go on with the American diet, they're the people who work in the nutrition area of FDA, I've gotten to know them pretty well. They are really hardworking people. And they're limited in what they can change by the laws that Congress passes. And again, I don't want to get into a back and forth about it. I would just say I want to stand by the people who work at FDA. They're good people, they're hardworking, and they want what's best for the American public. I have no question about that.
Now, and you seem very upbeat about the hiring. You were just talking about it. And so do you foresee any kind of mass exodus or change at the agency at the sort of the staff, the drug reviewer level based on what's happened in the election?
I think people are waiting to see. From things that have been said, the change could happen internally or for external decisions made by the administration. So I I just think we have to wait and see and have some faith that hardworking, high-quality people are going to still be in place, and we'll have support, I hope, from the external regulated community, which, given the dynamics, is likely to be the most important factor. I don't think there's a lot that people inside of FDA can say, and what I've seen already is that they're putting their heads down and doing their work.
Yeah. All right, you actually had you had some Republican support in your confirmation the second time around from Senator Burr, for example. But I'm curious, what kind of qualities would you hope for in the next FDA commissioner, and what kind of qualities do you think that next commissioner will have?
Well, I think I'd list a couple of very important ones. I think ability to function as an executive is going to be a very important one. I'll point to, this is not the first time I've ridden out of town after an election.
Yeah.
And I think Scott Gottlieb was like, you just could not have found a better person for the role at that time. And what I mean by executive function is the ability to listen to people with disparate points of view and take in and learn in a new environment and then make executive decisions. But right up with that, I would say, hopefully it'll be someone who understands that the really critical role of high quality evidence in everything that we do. And sort of related to that is a confidence that there is such a thing as expertise, because I feel like a lot of what's in question right now in our society is, should we make decisions based on how we feel at any given moment in time versus analyzing things and coming up with an opinion, particularly as it relates to issues that affect our long-term outcome. And the role of experts in that is really being questioned right now in a major way. And I'm not saying that experts are always right. In fact, experts are often wrong. That's been a big part of my career, is showing that by doing clinical trials that get very surprising answers compared to what experts would have predicted. But not having experts, I think, historically in every society has been a case for demise of that society. So I'd say those are the main things: executive function, a commitment to using evidence to make decisions, and really believing that there is such a thing as expertise.
Those are all good ones. There are concerns that FDA could be a target for sort of political interference from the White House or HHS over actual approval decisions. Is that something you ever had to deal with in your career, your 2 times at FDA, or was ever an issue?
Well, let me say a couple of things about that. First of all, it is very clear that there is a hierarchy of, it's an executive branch. So you got a commissioner, and you've got an HHS secretary, and you got a president. And the tradition in 99.95% of FDA decisions about individual products is that those decisions are made by career civil servants. The commissioner actually has no role in that unless there's an internal dissent and an appeal, or in some cases an external appeal that makes it all the way up to the commissioner level. But it's totally within the law for the president or the HHS secretary to overrule the entire FDA. And so that could happen. Did it happen to me? No. But I also want to say, I think the view that there's a sharp line between political and scientific, we got to speak truthfully about this. It's not a sharp line in many cases. I feel like for individual product decisions where you have high quality evidence, where the criteria is based on survive, feel, or function, the sort of standard criteria for medical products, there is no role for politics there. If a drug enables people to live longer and the benefit-risk equation is positive, by law, the FDA should approve it. But there's so many other aspects of this that are integrated in various ways with politics, and there's just no way around it. I have a whole series of essays I'm going to write when I finish. I'm up to over 90, but three of them are on something called OIRA, which I've been asking as I go around to public health schools and other places I've been doing lately because of H5N1. No one knows what OIRA is, but as I think you know, if FDA wants to put out a rule which is interpreting a law basically that Congress has passed in a way that has a huge impact on what happens, it cannot get through without OIRA letting it through. And that's a political arm of the White House. So to argue that there's no integration of politics and science, I think is a mistake, and we're going to need to talk bluntly about this, I predict, as we go through the next phase. There's a lot at stake for the cancer community and the industry.
I want to shift now a little bit to just very briefly on the Inflation Reduction Act and sort of the impact on drug development. And I was just curious if you've seen any evidence at all that drug development is changing in any way as it relates to the IP protections for small molecule drugs versus biologics, the fact that they have less protections. And is there any cause for concern based on that?
There's always cause for concern when you make major change, but I mean, you know that I'm on the record going way back. I mean, American patients have been gouged by the industry. There's no question about it. How can it be that you pay, like for, let's take the anti-obesity drugs now, 10 times as much in the US as Canada? I'm not sure that number's exactly right, but it's a lot different. This is not right. And so whether the IRA is the right way to approach it, I think there's legitimate cause for debate about that. But the idea that, what is it, like a third of cancer patients now are having very severe financial distress, it's not right. And so IRA is one approach. I don't think there's any doubt that it has affected the portfolio decisions that pharma is making. But I also don't think that anyone that I know of can say definitively whether that's better or worse. After all, drug development is so much like soccer. And, you know, I'm a Duke guy, so it's basketball, but turns out the men and women's team at Duke now are in the top 5 going into the playoffs. And I don't know, the ball goes all over the place, and a lot of stuff is planned, but when a goal ultimately gets scored, it's often because something completely unexpected happens. Somebody tripped up or, you know, things happen. And I think for drugs, I see no reduction in the innovation and creativity of the American industry. And I also see the same thing globally, with the U.S. clearly leading the way. And so what makes it through that sieve, I don't know. But I don't think it's right that Americans pay so much more for the same product as people living in other countries. It's not right.
Okay. And do you worry about how the public discourse on drug pricing has seeped into the FDA approval process almost? And I point to accelerated approval, where there's you know, the idea that prices are the same for something that had a lower bar of approval. I'm just saying that's the general discourse around it.
I have opinions, and then there's official policy, and those are not
opinions?
Necessarily all the same. I spent some time with the Israeli group that handles approval and essentially the CMS function. And I would really like it if pricing was gauged to demonstrated human benefit. And so, as you know, I'm a big fan of accelerated approvals, but whether that should command top price relative to a drug which has been shown to save lives, I really question that. And if we want to incentivize the industry to get that evidence, we're going to have to reach some accommodation on that, I hope. I mean, otherwise we're going to be in this situation. You know, it's also known I have a brother with pancreatic cancer who's getting excellent treatment, but I've also been able to see what happens out there, and it is like the Wild West. You know, what protocol you end up just depends on what place you happen to go to, and there's no particular order to it. And even for therapies that are on the market, there are all these different opinions, but very little evidence in the post-market phase in cancer. And yet most of the decisions that are made are not about what you start with, it's what happens after that now. But we have no system that's producing the evidence that's really needed. And the FDA can't mandate that. That's a whole other discussion. And so I think we need to work on that part of it.
Okay. And then, you know, on accelerated approval, we've seen such a large majority of those types of approvals have historically been in oncology. And I'm curious, do you see the potential for AA to really expand in a much bigger way in other therapeutic areas? And in essence, is it underused? Should it be used much more across the board?
I don't know that it's underused. I still believe, you know, in my area of ischemic heart disease, we have a lot of therapies that work, and the idea that you would put something on the market because it affects a biomarker, when we've had hundreds of treatments that look great from the biomarker perspective, it turned out not to have the benefit when it came to survival or prevention of serious events. I'm not sure that it's underutilized, but I think every case needs to be looked at, and I think CDER and CBER, as they have this new rare disease hub, you're going to see a lot of ferment of methodology of how to think about probabilistic Bayesian thinking. I would guess that almost everyone that's been involved in drug development, you know, I've been a trialist working in drug development for decades, would agree that you start with this massive uncertainty when you have a target and a drug, and the goal is to reduce the uncertainty over time. And the reduction of uncertainty may lead you in a risk outweighs benefit category, or benefit outweighs risk for various indications. And so it's entirely possible that in some common chronic diseases that don't currently use it much, there could be some good cases, but they need to be individualized.
Okay, I want to move to clinical trials very, very briefly and talk a little. You know, the trials are becoming increasingly complicated, resource intensive, time restrictive in terms of diversity of at times restrictive in terms of the diversity of the participants and the sites. Are there ways that FDA can really help identify the appropriate situations where that doesn't need to be the case, and what do you think can be done to address that?
Well, I want to commend Dr. Pastor and his group for working pretty hard on this, and this has, as you know, been a big part of my career, but it seems like the better the theory gets on simplifying and making trials pragmatic, the more complicated people make it, and I'm hoping that we'll hit some threshold where that changes. I do have a belief that in general, the pre-market phase, it could be made a lot more efficient, but that's not the big deal to me. As I've already said, I think the big deal is doing very efficient, streamlined, pragmatic clinical trials post-market to answer these questions that have a huge effect on clinical practice and patients, where, you know, what you get in that post-market phase is so dependent just on marketing, not on high-quality evidence. And I think that needs to be fixed. And the only way to fix it is to do trials at a lot lower cost. Now, if we learn how to do trials at a lot lower cost, which we used to know, by the way, we just need to go back to what we knew before, that could have some spill back into phase 3 to answer some of the dilemmas that, you know, we are uncomfortable to talk about, like you mentioned, diversity. No one wants to do trials that last 10 years to get a product on the market that saves lives. On the other hand, if you're going to study 100 people and put a product on the market, how can you even talk about diversity, especially from a global perspective? We have over 250 ethnicities in the world. 2/3 of the world's population lives in Asia. We have 4% of the world's population. So there are all these issues that can only be addressed if we enhance the clinical trials process. And I understand that's what the rest of this meeting is about. Did I get that right?
Mm-hmm. Yeah.
Well, I hope you really get down to it. What worries me is that remember, getting a product on the market in the United States is a decision by the FDA that the benefits outweigh the risks for one indication and one population. None of the other questions have to be answered, and FDA has no authority to make the other questions be answered. You don't know how it compares to other treatments. You don't know how long to give it. You don't know how to combine treatments. You don't know when this. You don't know the value of the treatment in terms of overall clinical outcomes. So we need a system that deals with that.
Okay, I have about 700 more questions, but I am not going to get to them right now. We have a couple more minutes, and I just want to get to some quick sort of questions here. And one of them is the idea of, we talked about sort of the chain of command and HHS. And I was curious how you feel in general about the center directors when they have to overrule staff on approval decisions, and when does the center director sort of get involved in that kind of process?
Well, I think in the ideal, I'm an observer of this. I've never been a center director. I've never been a staff. I was a deputy commissioner for a while, so I'm just an observer. But this is part of what I mentioned with executive function. In the ideal world, as I said, that uncertainty is being reduced. I think the most underappreciated part of the FDA is all those meetings that occur before the final application goes in. And during the course of those meetings, if there's a controversy in the ideal circumstance, the staff and the leadership of the center would be in conversations to resolve those issues, so overruling would be unnecessary. So in the ideal world, you should never have to overrule your staff. But we don't live in an ideal world, and we have personalities in the FDA, and we also have a situation, in a way, it was sort of the good old days when I was on the cardiorenal panel. There was one FDA opinion. No, it was opaque what all the different sub-opinions were within that. Now, everyone can express their opinion in writing, and so that leads to much more give and take between the center director and the staff. So, but I think a goal of any center director would be to make it so the goal should be sort of like ventilator-acquired pneumonia. 0 should be the goal. Gotcha. Realizing in the real world you can't quite achieve that.
And then I think lastly, we have about a minute or so left. And what do you want to accomplish in your remaining few months as FDA commissioner? Do you have 2 or 3 priorities that you have set on, or what are sort of the few things that you want to get accomplished in the time you have left at the agency?
Well, I feel like the medical products are on the way, and there's some very significant, relatively technical things that need to get done. The big deal for the public health is tobacco and food. And we have major things there that we're hoping to get done. It's publicly known the menthol rule has been an issue, and we also have a very good understanding of what the addictive level of nicotine is. So the nicotine standard would be the biggest deal. We can't enforce a nicotine standard, but we could possibly put out a proposed rule that would unveil to the public what's at stake in terms of public health. And then for food, it's just simple stuff. Like, how can it be that you can't get the label on the front of the package? That just sounds crazy to me, but it's an ordeal. Many in the industry are opposed to it. And we're trying to update the term healthy so that when you put, let's face it, most people are busy. When they go shopping, they don't study the label and take 10 minutes for every decision. They react to what's in front of them. So if we could just label things that are basically healthy as healthy, that would make a big difference. Those are the very most important things. I don't want to belittle the medical product side, but the changes there are technical and very important to the elements that are involved. But food and tobacco, that's what's causing the chronic and serious crisis we're having right now in our life expectancy.
All right, well, thank you always for your honesty, Dr. Califf, and you've done a really terrific job as FDA Commissioner, and thank you so much for this session, and we'll move on to the next session, so please.
Appreciate the chance to be here. You guys, you have to fix the pragmatic trial issue. It's so important.
Pragmatic trials.
All right.
Well, thank you very much, Dr. Califf. That was a strong charge for one of our upcoming sessions. But thank you for being here and for your continued leadership. And Ramsey, thanks very much for leading the discussion this morning. As we get started with the first panel today, I have the job of giving some instructions, so please bear with me while we, I think, transition the stage here. I'll remind everyone that's participating, either in person in the room here or virtually today, that you can add to the discussion by following us on social media via LinkedIn or X and join the conversation using hashtag Friends Annual Meeting, Friends AM24. And as you can see in the meeting agenda for today, we'll be starting with 2 sessions this morning, followed by a lunch and keynote discussion with NCI Director Dr. Kimran Rathmo, followed by our final session of the day. For each of the sessions, all the advanced registrants, you should have received a copy of the white papers that were prepared by our expert working groups throughout for the discussion of each of the sessions today. They'll be guided by those papers, so hopefully you had the chance to read them. If not, you can also access them with the QR codes that are on the meeting material today. As you'll see coming up in just a second here, each of the sessions today will feature a lead presenter to frame the topic at hand, followed by a panel discussion based on the topics in the white papers. We also want to encourage and provide the opportunity for audience participation in the discussion, so when it's time, you'll be able to find microphones in the aisles, and we ask that you briefly introduce yourself and your organization and strive to keep your questions or comment on topic, please. For those of you joining virtually, please feel free to use the Q&A function in your Zoom bar, which will be monitored in the room today, and we'll be able to pass those questions up to the moderators for the panel discussion. So, without further delay, let's get started with our first session, which will provide insights into conducting interim overall survival analyses and clinical trials. And to start us off for that discussion, I'm pleased to introduce Dr. Jeff Oxnard, who is the Vice President of Clinical Development at Eli Lilly and Company. Jeff?
Hello, I'm Jeff Oxnard. I lead lung cancer clinical development at Lilly. I was last at this session 6 years ago when I moderated the kickoff of the ctDNA project. That was back in my days in academia. Today I'm going to speak from a drug development perspective on interim OS, and I want to thank Jeff and Mark for inviting me to talk. We are lucky to live in a time when there's been dramatic improvement in our cancer therapies. We have a wider range of therapies available for our cancer patients. And as patients live longer, that means that in many cancers, OS no longer can serve as the primary basis of a benefit-risk assessment. And intermediate or surrogate endpoints are increasingly adopted as trial endpoints for assessing risk-benefit. I'm going to do a quick history on this. On the left side of your screen is the first trial I remember as a trainee that's adding bevacizumab to chemotherapy in first-line non-small cell lung cancer. No one remembered a positive trial in lung cancer before that. And then with that OS hazard ratio of 0.8, the standard of care changed. When you look at the label of bevacizumab in lung cancer, the overall survival is shown. The PFS is not relevant for a drug where there has been no progress. But 6 years later, this is erlotinib in EGFR mutant lung cancer on the right-hand side. There are more therapies in lung cancer in first line and second line. This trial showed a dramatic improvement in progression-free survival, but actually no difference in overall survival. Patients went on to receive different therapies subsequently. This trial was approved based on progression-free survival. The label highlights the progression-free survival difference in the overall survival is used as context. I'm speaking about the right-hand side today. Therapies that demonstrate a meaningful improvement in a surrogate endpoint where OS nonetheless is collected and used to contextualize that surrogate endpoint difference, to look for benefit risk or to help understand harm safety. But that interim overall survival can be challenging to interpret at the time of the initial trial readout. It is generally immature. It can be unstable over time. And interim OS is especially confounded by crossover in subsequent therapies. And so the question becomes, how can we as drug developers and a cancer community best analyze and interpret this interim OS data? For interested readers, I want to highlight this fantastic paper. It's a required reading with a statistical perspective from a recent ACR, ASA, and FDA workshop. I thought this figure was especially interesting. On the left-hand side, you see a trial where the overall survival curves play out in a predictable fashion with proportional hazards. But in the middle, you see an example of a trial where the curves cross, where early on, one arm seems to be doing better, later on, another arm seems to be doing better. Classically, immunotherapy can behave with this in this pattern where patients can initially do poorly and then have dramatic benefit. The point is that overall survival is not always predictable based on early looks, interim looks at survival. But what I'm going to do is use a couple drug development anecdotes from a Lilly perspective where we had trials with positive surrogate endpoints where we had to then consider how to use overall survival and other information in an interim fashion to contextualize the findings. The first trial is Monarchy. This is a trial in adjuvant breast cancer, adding a CDK4/6 inhibitor to hormonal therapy in hormone-positive breast cancer. This has a long natural history. Breast cancer patients can live a long time, and that means overall survival can take a long time to play out. What you see in the curves from the publication is that the primary surrogate endpoints, invasive disease-free survival, distant relapse-free survival, were positive and were meaningfully different years 2, 3, 4 into follow-up. And yet overall survival is pretty much overlapping. There's no difference in the overall survival early on. Now, these patients with breast cancer are developing metastatic recurrence at a different rate. You presume there will be some difference in survival over time, but you don't see that early on. And when the OS hazard ratio is actually 1, then at any given moment, statistically, your hazard ratio is going to look like it's above 1 or below 1 about 50% of the time, right? Just based on how the events are playing out. And so it's hard to interpret how the OS will look at those early time points. In fact, when you look at the readouts from this trial, at interim analysis 1, you see a positive hazard ratio, a meaningful difference in IDFS and DRFS, but the OS hazard ratio was 1.09. And yet 15 months later, the surrogate endpoints have about the same hazard ratio, but the OS hazard ratio is 0.93. Now, there's no statistical difference between these numbers. They're both kind of overlapping broad confidence intervals, hard to interpret, but we gain different confidence if they're on one side of 1 versus the other side of 1. And to some extent, the question becomes, do we as a community want to act urgently when you first see the difference in a surrogate endpoint? Or do we feel less urgency and want 50 more months to go by or more before we have confidence in the risk-benefit from that data? I'll just say, in the FDA label, this was eventually approved, and you'll see the positive IDFS hazard ratio, the curve that I showed you, and the OS, all that is said in the label is, at the time of the interim analysis, it was immature. Only 6% of patients had died. Because fundamentally, this OS data is still too immature to have a meaningful inference to provide to patients and physicians to understand this. All of this is sort of behind-the-scenes context to understand the data. I'm going to give 2 more anecdotes. The next is RET inhibitor therapy versus chemotherapy in RET-positive lung cancer. This is a study that enabled crossover. And what you see is the PFS hazard ratio in this New England Journal of Medicine publication last year is 0.48, a very positive trial for the surrogate endpoint, but the OS hazard ratio is 1.04. And what can happen when there's crossover, it can create some of this disconnect between the PFS and the subsequent OS readout. Just looking at the trial in detail, when a patient was randomized with RET-positive lung cancer to chemo with or without immunotherapy versus RET inhibitor therapy, they were told that if they were randomized to chemotherapy, they could access the RET inhibitor at progression. And that motivates enrollment into a randomized trial. There were very rigid criteria about when they could be crossed over. But what you see is that those patients who get chemo first, then RET inhibitor therapy, receive 2 high-quality lines of therapy. And yet patients on the RET inhibitor, they don't have crossover. And in fact, if you're progressing on the pill and you've now got progression, do you want to switch over to chemotherapy or just take more of the pill? And a lot of the patients actually just stuck with the pill. They didn't cross over. And there was an imbalance in how much therapy was received on the 2 arms. When you look at the eventual FDA label, here's the positive PFS. This was recently updated. And I just want to read the overall survival to you. The overall survival was immature at the time of the interim. At an updated descriptive analysis, the OS hazard ratio was 1.26 with a broad confidence interval, and it may have been affected by imbalance in post-progression therapies. Of the patients on chemo with or without immunotherapy who progressed, 74% received RET inhibition at progression. But of patients on the RET inhibitor who progressed, only 23% chose to go on to chemotherapy at progression. And actually the majority just continued the RET inhibitor. That imbalance of sequencing of therapies means the overall survival is pretty hard to interpret. It's in the label for context, but understanding that context is pretty confusing for most to use. One more anecdote. The final anecdote is RET-positive medullary thyroid cancer. This is a disease with a long natural history. And actually, when this trial was designed, it was designed to change PFS as a primary endpoint. And you see PFS hazard ratio 0.28 in the New England Journal of Medicine publication. But overall survival is so prolonged in this disease, actually OS was not called out as the key secondary endpoint, because the theory was that OS would not be able to be impacted. Actually, when you see the OS, there was a small impact on OS, and that's actually played out more over time. And so maybe we predicted that wrong. But instead, the key secondary objective was called comparative tolerability, a quality of life endpoint. This was envisioned as another tool to contextualize a surrogate endpoint. And it was pre-specified as the proportion of time on treatment with a high symptom burden as assessed by FACT-GP5, and this was alpha-controlled, p.025. And what you see in the ASCO data shown here is that patients on the RET inhibitor had a lower proportion of time with high side effect burden. Patients randomized to the multi-kinase inhibitor had a higher proportion of time with a high side effect burden, and this played out over time. When you look at this data in the label, again, you see the progression-free survival. OS was commented as being immature at the time of the analysis. And on the right-hand side, this is that quality of life data now appearing in the label to contextualize the progression-free survival surrogate endpoint with a descriptive difference in the portion of time with a high side effect burden. I'm going to end the didactic period and ask a couple questions that motivate our upcoming discussion, okay? And the questions I take away from data like this is, in a trial with meaningful differences in PFS, DFS, how should we, as a scientific community, interpret interim OS? Do we feel an urgency to act on a positive PFS, DFS signal, even when the interim OS is immature and hard to interpret? Are there best practices that we can be using in our trial design, in our statistical methods, that maximize the chance of an interim OS being informative when a positive trial reads out? And finally, are there other metrics that we could be using, like quality of life or other, to inform the risk-benefit of a regimen when OS is uninformative or confounded at an interim time point? And with that, I'm going to introduce Dr. Keith Flaherty, who is going to moderate the next dialogue between some folks on the panel. Thank you all. I'm going to sit.
All right. Thank you, Jeff. And thank you to Ellen, Jeff, and Mark for keeping me coming back. I'll never say no to this meeting, that's for sure. I'm a medical oncologist at Mass General, been there for 15 years now, at Penn for 10 years prior, focused on therapeutic development in the academic environment, and have co-founded a handful of companies. And from both perspectives, I'm deeply interested in the question we'll delve into today, as well as pretty much all the questions that Friends of Cancer Research asks us to meditate on. So I'd like to introduce our fellow panelists. Jeff Oxnard, you've met. A couple of our other panelists began this discussion with Friends of Cancer Research and other colleagues in the field last year, including Nicole Gormley, Division Director, Division of Clinical Hematology at FDA, where Nicole kind of has a broad view on these topics with both clinical and statistical expertise in bringing such reviewers to bear on discussions in terms of OS-related issues. We're thrilled to have David Mischel with us, a patient advocate who's been very much living on the cutting edge of early access to therapies for 14 years now on continuous therapy for multiple myeloma. Reflect on that number and the number of approvals that have occurred in that time frame. David's currently on a 4-drug regimen, 3 of which came to market under the auspices of accelerated approval. So an incredibly remarkable journey, and we thought kind of brings an incredible personal perspective. But David has also been lending his time and insights to ODAC and has been participating there as a consumer representative. Lisa Rodriguez, also a Deputy Division Director in Division of Biometrics 9 at FDA, has also been participating in this ongoing Friends of Cancer Research supervised discussion of interim looks at OS. Lisa's a statistician and deputy division director in the Division of Biometrics 9 at Cedar. They're reshuffling, interesting. Don't know why they're doing that, but okay, I guess there's some logic to it. And then last but not least, okay, now you're in order as I'm introducing you now, I'm figuring that out. Tony Sabin, who's head of immuno-oncology statistics at GSK. Tony has a 30-year career under his belt in the pharmaceutical industry, currently focused on immuno-oncology related matters, which, as Jeff touched on, is one of the areas that we think about in terms of important context here for this discussion about early looks at interim analysis. So with that, just to orient you to the session, we have about 40 minutes that we're going to sort of review and look forward on the discussion that we've been having on this topic, and then hold the last 10 minutes of the session for Q&A. As always with this meeting, we will give preference to in-person questions, but are open to questions coming from online participants as well. So I'll indicate when we're sort of transitioning into the Q&A time, but certainly please start thinking of questions up to that time, and feel free to step up to the mics when we get to that session. So I will take my seat now, and hopefully my lav is on. Okay, so Nicole, if you would be so kind, maybe you could just kind of give us your perspective on how we got here, which is to say, kind of what are the developments, recent trends in terms of trial results, certain therapeutic indications, certain therapeutic modalities, that kind of bring up this issue of trials that are coming forward with NDAs based on earlier endpoints, progression-free survival, durable response, whatever, but where OS interims have become increasingly important in the eyes of FDA.
Yeah, thanks. So great discussion, introductory presentation, and I enjoyed the discussion at the last session as well. So, in terms of how we got to where we are, overall survival has always been a key endpoint that we have cared about in oncology. And I think we, over the past recent years, have had just such improvement with our therapies that we have gotten to a point where we can't rely on it as a primary endpoint. So in my division, we review products for multiple myeloma, lymphomas, CLL, and these are diseases where, take multiple myeloma, for example, it's now thought that patients that are newly diagnosed currently will have an overall survival approaching 10 to 15 years. And we've had over 20 therapies approved in the past 20 years, which is quite impressive. And they are very effective therapies that, thankfully, have resulted in great long-term outcomes. But as a result, we can't rely on overall survival in that disease as a primary endpoint. That being said, we always look at overall survival because of its importance and its role in having confidence in the risk-benefit of any given product. We've also found ourselves in a situation recently with several therapies where we've seen at times discrepant results between the early endpoints and later endpoints. But in general, we've gotten to a point where we really need to be able to rely on earlier endpoints in certain diseases to be able to approve a product. And so still needing to look, though, at that risk-benefit assessment and looking at the totality of data, including an assessment of OS. And so that really prompted us, you mentioned earlier the workshop that we co-hosted last year with AACR and the American Statistical Association to look at overall survival as a safety endpoint. Are there ways that we can pre-specify the assessment of overall survival from a safety perspective that would provide meaningful information when we're using earlier endpoints to make a regulatory decision?
Great, thanks. So we are going to get into the weeds here of that discussion that began last year in terms of statistical designs and also just analysis, sort of analysis plans prospectively designed to try to accommodate this issue. But before we dive into those weeds, maybe Jeff and David, each of you could kind of reflect on how this discussion is, influencing your thinking about trial design. Jeff, you gave us some really nice recent historical examples, but now how might one think about designing a trial, to, I don't know, provide more information, basically, and regarding this issue of OS when it's a secondary endpoint. That's obviously the context that we're wrestling with here. In terms of, shifting analysis timelines, delaying analysis, for example, of even an early endpoint like PFS, so that OS is a bit more mature by delaying crossover. I mean, there's multiple levers to think about, but how do you think about that?
I think there are healthy things a drug developer can do, like make sure you collect the OS. Just remember that OS is collected by the site, who has to follow the patient after they've stopped the therapy and are no longer on treatment, right? And so there are some sites who aren't that motivated to keep following the patient. So there is sort of how do you design the study to effectively not lose patients and collect that OS and don't let them withdraw consent. So there are some operational details that we, as a research community, need to commit ourselves to collecting this information, and that's achievable. I think there are also some more maladaptive steps the developer can do. I am surprised how often crossover is not included in trials that I would think would be a crossover trial. And I think having seen the example that I showed you before, right, I can understand how crossover might create a kind of uncertain jeopardy in, wait a sec, how will my OS play out? It might be good for patients, but it's not good for my program. We're just gonna not do crossover here. And again, it is allowed to do crossover or not. So I guess I'd rather there be more good steps we can take in this environment, but I'm worried there might be some ill steps developers are taking as well.
Yeah, thanks. David?
Well, first of all, I want to emphasize I'm a big fan and beneficiary of accelerated approval. Drugs that would not have been available to me for maybe 2 or 3 years until 2 or 3 years later were there. And if it's 10 to 15, I'm pushing it at 14 years since diagnosis this month. So I want accelerated approval to happen. With confirmatory trials subsequent to approval to make sure, especially on OS, that we don't have a safety problem. And we saw that the lines in your presentation can cross, over time. So overall survival continues to be, from my perspective as a patient, a very, very important measure. And I think Dr. Pazder has indicated that the FDA would be pleased if the drug companies started their confirmatory trial at the same time they start their accelerated approval trial, so that they could populate that study and have it be in place. And that's going to lead to more, if the companies do that, more interim OS data available. But interim OS data is scary to me because it has so many potential limitations. And I don't want drugs delayed because of immature OS data, interfering with them getting accelerated approval. And I don't want drugs approved or based on immature OS data that might help lend some belief that what we see in the surrogate endpoints, reasonably likely to predict clinical benefit, that immature OS data shouldn't trump the endpoints that the study was aimed at. And so I think it's very, very important, and Dr. Gormley can talk more about this, that the FDA, works with drug companies through the development process and helps the drug companies decide what the FDA will consider to be persuasive and whether they want to bring forward immature data that could, in fact, get in the way of approval. So I think we have more to talk about, a lot more to talk about how to do that. And then finally, a point on, for example, multiple myeloma and OS overall. Once you get through induction, they put you on combinations. I'm on 4-drug combination. And Dr. Califf talked about the clinical variation that exists around the country in how they treat patients after induction, where there's kind of a standard of care, but even that varies from major institution to major institution. It's very difficult for me increasingly to conceive how you do trials because they're doing so much behind the initial approval that you can't run a tight ship because they're doing this over here and that over there, and it is very, very challenging, which is the point you were making about OS over the long haul. It's a good problem to have, by the way, as long as all the patients are getting therapies that work. But the clinical variation is still remarkable for myeloma, and that's from places like Dana-Farber to the Mayo Clinic to Memorial Sloan Kettering. You go to, and even within those institutions, different physicians practicing in different ways. So OS becomes more difficult to use, and yet for me, it's a critical safety measure.
Yeah, thank you. So it's, at the risk of, making this conversation seem like it's, kind of equally relevant to all contexts, I do want to maybe try to dive into some specific examples. I mean, if you think about all cancer types, regardless of, let's say, spectrum of aggressiveness, I mean, glioblastoma, pancreatic cancer on kind of, a far end, chronic lymphocytic leukemia, maybe, in the other end, one can think about contexts like that where, you could imagine even a relatively, seemingly big effect on response rate, progression-free survival, being anything but clear at the level of overall survival, again, CLL under the spectrum. We expect that there should be, kind of consistency of endpoints in the more aggressive disease, limited option circumstance. But Jeff, if maybe you could just grab an example from your presentation and sort of maybe talk through, kind of the clinical interpretation of a conflicting result, where response rate is seemingly promising, PFS, but OS is, hazard ratio above 1, which is that, just to be clear, that's the scenario we're talking about. Anything below 1, the lower the better, obviously, but still, it's really this, early result above 1, like it's kind of how you think about the, again, clinical interpretation in, a given context like you discussed.
Sure, and it's not just above below 1, right? We always want the OS to follow the PFS. And when you don't see that, I think any investigator or developer is going to say, what's going on in my data? The obvious thing you're going to look at is adverse events that make a efficacy signal, outbalanced by some interim mortality. Are you seeing high-grade fatal events, right? You're looking for toxicity. That's the clearest way to measure harm. We're talking about harm, right? Are patients having fatal infections on one arm that is eroding the survival potential of a progression-free survival impact? In the absence of seeing grade 5 events and other toxicity that explains this disconnect, that's when you start looking more at the statistics and saying, wait a second, like, what's the shape of this? How could it go over time? What's the width of the confidence interval? And then I think we start looking at variability in subsequent care as you described. And when you start seeing variability in subsequent care beyond your PFS, then you start saying, wait a sec, I don't know what this number means at all, given all these other things contributing. The exercise I just described is really what we want, I think, all sponsors to do. Please be vigilant about safety, be vigilant about harm, look at dangerous events. And if everyone's doing that diligence, we should have more confidence. In the end, it doesn't change the hazard ratio of your interim, and you still have to have a number in the end that you need to do something with.
Yeah, Nicole, maybe you could reflect on that kind of same perspective on the kind of receiving end of data to the degree to which you can kind of unpack some of these questions of like what might be contributing to harm when when looking at this kind of a hazard ratio above 1 situation.
Yeah, and I think that's exactly the situation is sort of what we're trying to, in a sense, trying to avoid. by at the outset of the trial, sort of understanding a little bit more about what are the likely contributing factors. Are we likely to see crossover? Are we likely to see, are there a lot of subsequent therapies? Are we likely to see non-proportional hazards? And I know we don't always know that a priori, but if we are have other information from previous products within the class, using that knowledge to inform our trial design and what we pre-specify. And so, one of the important factors is just the concept of, and one of the discussions that we engage in with when we have conversations with sponsors when we're talking about a trial design is how much overall survival information do you expect to have in various time points throughout the trial. Specifically, at an earlier interim analysis, how much information do you expect to have? How much information regarding overall survival information fraction will you have at the time of your readout for your accelerated approval endpoint? And then how much time until you'll have more complete or a final OS assessment, if it's powered for that sort of assessment. And it's important, what we're talking about is that we've always looked at overall survival information because it's part of the entire data package, the totality of information. And so we're talking about having more rigor around what is pre-specified to inform decisions at a later point. And even at earlier points, right? Some of the examples that we've had within this disease space, I'll mention the PI3 kinase inhibitors, those were interim assessments of overall survival, and they were really, in fact, very early analyses, even before, really what we're talking about. But they suggested so much harm with, overall survival hazard ratios of 2 and 3, and that coupled with the safety information, which was showing significant toxicity and discontinuations and dose modifications needed for, to be able to remain on therapy, really sort of provided valuable information. And so I think at any point when we're meeting with sponsors, we're talking about how much information will you have at each time point, and then that's always in the context of all the available information.
Great, thanks. We're promised just about to dive into the weeds, but David, just before we do, for an agent that makes it to market with a, let's call it ambiguous result of overall survival, I mean, do you have concerns in terms of, how doctors and patients, kind of receive, label information regarding, this, like, again, immature OS, but like this, the degree to which that can be sort of understood and communicated, it just, again, clinical practice.
Well, I don't want the FDA to make approvals based on immature OS, interim OS. I'm afraid that we could err on either side. It can be useful to create that context that Dr. Gormley is talking about, but so are, AEs where you get dose modifications, dose reductions, or you see things happen to quality of life, but measurable and with rigor. I'm not a big fan of anecdotes. All of those things figure into the picture, but the FDA and the sponsor have to reach an agreement on whether or not interim OS data belongs in the picture, because it may not. It may be inappropriate. It just may be too small, too immature. We shouldn't be drawing conclusions from it.
Yeah, thank you. Okay, Lisa and Tony, take us down into the rabbit hole here. Lisa, can you maybe reflect on last year's discussions and ongoing in terms of pre-specifying endpoints and approaches to trying to understand these issues of conflicting results?
Yeah, so last year's AACR conference or workshop, I think we did talk about a lot of aspects that would be important to designing the studies, considerations for everything that was talked about in terms of what sort of data you might be able to expect. So certainly at interim, when you do an analysis of your primary variable, which may often be PFS, we would want to see OS. Now, yes, understandably, there may not be much information, but I think prospectively thinking about this, and as this was mentioned, the sponsor understands their drug the best. I think they can really be thinking about what is feasible, what's possible, what's going on with this patient population in particular, do some projections, do some calculations. Certainly simulations could be a part of this. What are the potential patterns that you could be seeing? What is the potential amount of information? And really be thinking about how you're going to assess risk, how you're going to assess safety, what your definition of harm would be, and thinking about that prospectively. I think the more that sponsors are able to bring to early discussions with the FDA and at the IND stage, designing your study, we can have a more robust discussion about that information, what's possible for possible use for decision making at different time points.
Great, thank you. So Tony, then following that, kind of how are you, from a sponsor perspective, thinking about kind of various options in terms of how to design and lay out a statistical analysis plan that kind of accommodates these concerns?
Yeah, so first and foremost, I think as Dr. Gormley really described well, I mean, you can't stress enough, I think, the importance of good, robust trial designs to try and mitigate this as an issue from the onset. I think once you're faced with those results at hand and OS is immature, and again, decisions are naturally going to start by focusing on the clinical context and the totality of evidence. Statistically, though, where if data permit, I think in addition to your typical interim analysis type frameworks, often group sequential type frameworks in oncology, and estimating the hazard ratio and the confidence limit for that hazard ratio, and perhaps interpreting that with respect to some threshold for harm. There's a few other pieces we can do, for example, the Bayesian predictive probability, Bayesian predictive modeling, which attempts to try and predict what your OS is going to be at your final analysis. There's a variety of different treatment switching methodologies, and lately I think we see a lot of discussion about tipping point analysis, whereby you're really trying to test the robustness of those interim analysis with respect to potential future risk of detriment. So I think there's a number of tools and options available there. I think there's some work to do probably to find the best way forward and help contextualize the use of some of these methodologies in this space.
Great. Jeff, you talked about, use the term maladaptive. Maybe you could expand on that a little bit in terms of, kind of what would you see as examples of, maybe call it overreactions in a way in terms of designs in light of this discussion?
I mean, I certainly appreciate the statistical expertise in being able to do this right. What does wrong look like? I mean, I don't want to be a role model for wrong, right? But you could imagine a trial over-enrolling and overpowering, hoping to have extra events for an interim OS when really what's meaningful is the primary endpoint, and that over-enrollment is not necessarily good for patients, and it might not be good statistically either, right? To some extent, when there's uncertainty in how the data is going to play out, that can create sponsors to make mistakes, right? And so I think the more we can create some conformity in what we think good looks like, the more we can all aim to achieve good.
Yeah, Lisa, it seems like crossover is a major issue at the center of this conversation because, of course, for investigational agents of unknown risk-benefit, the quote-unquote need for allowing crossover is hard. It's hard to make that case, and yet it confounds the results, one way or the other, and difficult to unpack in retrospect. But as you and your team think about trying to, kind of reflect on this issue, I guess both prospectively as you're meeting with sponsors, and kind of talking about a design for a registration-enabling study, as well as on the back end analyzing the data. Just maybe kind of, how do you think about this issue of managing crossover?
Are you talking to me?
Oh no, Lisa, I'm sorry. Staying in the weeds here for a moment. Yeah.
Yeah, certainly crossover can make it difficult to interpret. It could dilute the treatment effect. Certainly there's reasons to design your study with crossover. I think that should be carefully thought about and planned for. Perhaps have a discussion again with the FDA about this design. I think certainly prospectively we still need to plan for what is your primary analysis and then what are some additional analyses you can do. Typically we will look at the traditional analysis approach in terms of evaluating the hazard ratio, but as was mentioned, there's other methods that can be used potentially to account for crossover. Many of these do have some unverifiable assumptions, but certainly they can be used for supplementary analyses. I think a variety of analyses, looking at survival data in different ways, can certainly help us assess the robustness of the results. We certainly have concern as well at the patterns that you might be seeing in the data in terms of if anything is potentially informative. Are patients crossing over early, or are they crossing over at a certain point that we can interpret this timing as somehow informative? But I think the best approach, again, is prospective planning, a variety of supplementary analyses, discussions with the FDA. I think we've had a lot of discussions about the results. We're certainly open to looking at a variety of post hoc analyses, but again, that's to help the interpretation, not necessarily the primary analysis.
Right. Right. So Tony, again, back to the sort of getting ahead of the issue in the first place. Maybe you can reflect a little bit on this issue. Crossover is just, I think, a ready example to consider. Jeff was talking about one of his examples, recent examples. I'm mindful of the fact that really for a lot of the orally available, so-called targeted therapies, oncogene targeted therapies as a class, there's been a lot, and immunotherapy actually, I guess maybe could be included in this. A lot of post-progression therapy, has been allowed / occurred, usually after conferring with sponsor. But it's an important point to reflect on because it creates itself kind of this additional confounder, and one could argue keeps patients from moving on to another therapy, where a subpopulation of patients might actually derive benefit, right? And this it's an indirect form of harm in a way, and so trying to unpack this kind of the layers of confounding, I guess, is challenging. But you alluded to this before in terms of kind of design considerations, sort of time-based analyses for early endpoints versus OS, performing simulations, and how much can one get ahead of this in terms of a priori designs to try to you can't exactly predict the future when it comes to things like, okay, well, what fraction of patients are actually going to cross over, take advantage of that option. Might be different in the U.S. environment versus other global drug development venues or trial conduct venues. So just, again, knowing we don't have a crystal ball for all these things, how as you, as a statistician, how do you try to, prepare for these in terms of analyses?
Yeah, so, I think obviously, as we're alluding to, really a really key approach here is the use of clinical trial simulation. So in addition to evaluating your normal operating characteristics, your type 1 errors, your power, there is the potential here to look at what's the probability of observing some threshold for harm conditional on you being successful in your short-term immediate endpoint. I think if you can do that, this sort of approach could help sponsors and the regulatory authorities trying to balance what is a reasonable threshold for harm with ultimately the timing of that interim analysis. The longer it takes, the more information, the more precise the better the decision in some ways. But I think when you think to the crossover and the non-proportional hazards, well, naturally you can incorporate some of that into the the simulations, but you have to make some assumptions. And it's a lot easier now in the IO world that so many drugs have read out. So that makes life easier to do that. It's quite hard to do, of course, and predict with your first indications. But when you move then from out of the design and into the analysis, this is where it takes you back, I think, still to the predictive, the Bayesian predictive, perhaps parametric models, and combined with all the crossover methodology that we've got at the moment.
Great. So maybe Jeff, ask you to reflect, and let's move the conversation up, kind of this higher level beyond individual trials. Like, what can we do as a field to try to create sort of a stronger evidence base, understand a bit more about features of a novel therapeutic or in a given context that would allow us to get kind of better at getting ahead of therapies that end up actually having a harm, which is that's what this whole conversation is ultimately about. Is imagine the PD-1 example in multiple myeloma, where early analysis of overall survival showed clear, statistically significant harm, but we're talking about the unclear ultimately becoming clear, right? Getting ahead of that circumstance. Anyway, if that's where you're going, great. Otherwise, I'll ask Lisa and Nicole to help us think about this at a higher field level.
Two things, Keith. I wanted to react to your prior comment about post-progression therapy. I was actually a huge advocate of that back in my early academic days, right? Patient has no therapies left, continue the therapy, it's still probably doing something. But we're developing drugs in a world with more therapies. And so that approach that's very patient-centered, yes, keep going, here's the drug, risks becoming a harm. And that's an interesting instance of harm. It's not during your PFS, it's post your PFS. And I do think we have to wake up to the fact that with a world of more treatment options, we could be unintentionally doing harm by propagating post-progression therapy indefinitely. And that, again, as an early advocate for that approach, that settles a little bit ill with me. Certainly I know patients appreciate the momentum of sticking with the drug that's working. And as a clinical trial, maybe that's not the right thing. You raise what other tools or what other things can we be doing? Nicole, what you said earlier about an OS signal with these adverse events sort of clarifying it, that helps me think about really how can we use adverse event data in a richer way to characterize what we're seeing. I always feel like grade 2 events are really burdensome to patients, or grade 2 AEs, but they're not part of necessarily the regulatory speak about how a drug is effective or what risk-benefit looks like. I think a OS hazard ratio on its own feels like a very naked number that doesn't have a lot of meaning. And if we can more richly contextualize it with adverse events, quality of life, other things that I think bring to life harm in a way that's patient-centered, it might give us more information in addition to the statistical steps that we're talking about.
So I would just add, I think we've always been talking about what we need to have is a more statistical, rigorous assessment of OS, because right now it's just well, it looks okay, or yeah, it's going in the right direction, like a more rigorous assessment of OS, but it's always been in the context of all of the data that we have available to us at that time. It's just one more data piece that's rigorously assessed. It's not all or nothing, it never has been. So I do think it's important that we have a little bit more rigor around the OS, but again, it's always in the totality of all the data available, including safety and tolerability and PK information, PD information, et cetera.
So Lisa, the FDA is, more so than any one sponsor, sees effectively all the data, or the largest amount anyway. I mean, I suppose that's not true for a negative trial where no NDA is filed. You don't get that data. So like in your, in the context of, FDA and the data that accumulates, there over time for reviewed/approved products, or looking even beyond to data sets that aren't submitted for review. What would be your kind of ideal path forward, for the field to try to again better understand the predictors of therapies that turn out to actually have genuine harm versus those that don't?
And that was me.
Oh, that was Lisa.
Lisa,
okay.
sorry.
Well, and then back to you, Nicole, because I do want. Yeah.
I think the white paper that was released for this conference was. Had some good ideas in terms of the appendix, I think, talked about some approaches doing simulations and evaluating the hazard ratio in a variety of circumstances. And I think that's important to really think about. So I think in each of these cases, when we talk about how do you interpret the hazard ratio, it always depends, right? So it depends on the clinical context and a variety of things. So I think being able to look at what we're able to evaluate in terms of overall survival at different information fractions, perhaps in different scenarios with or without crossover, in different disease areas, and different expected levels of maturity by the end of the study. Certainly we can see a variety of circumstances and what are you getting, how are you defining harm in each of these situations, and what can you do if maybe at the different time points where you might do interim analyses, and how much more might you need, how might that change your conclusion. So I think really looking at that in a specific framework and looking at all of these different scenarios, I think that can be very helpful, and to be able to interpret those results in those different settings.
Great. Nicole, back to you.
I think Lisa said it perfectly. The only thing I would add is just that, like, we do have a lot of knowledge when we're talking about, designing a trial. And granted, we don't know everything, but there's information from other products within class. There's information from the earlier stages of drug development that really can help sort of inform appropriate trial designs and considerations then for pre-specifying the information. So just trying to take advantage of all that we have, I think, would be really important.
Great. We're going to move to questions in just a moment, but maybe if I could ask each of you to kind of reflect on a quick takeaway, coming from last year's conversation into this year's, in terms of this issue and kind of what you think is the most kind of pressing opportunity / unmet need to try to kind of better address this concern. Maybe you just go run down the lineup here, Jeff.
One thing you can always do with an interim result is wait for more information. And I'd like that not to be the answer. Like, in the end, we're trying to talk about drugs that we think there's some urgency to move forward. And so how can we find a way that's not just, well, give us another couple more years and then we'll know. I hope there's a different answer beside that.
I think my takeaways, I've sort of already said them. I think, pre-specification, and I just really want to sort of highlight as well, like, this is a really complex issue. There's so many factors to consider. Prior information we have, knowledge we know about the disease setting, other therapies that are approved, and then all the information that's known about this specific product. And so those discussions, I think, are and that information it's really should be considered on a case-by-case basis. And so I really encourage sponsors to come in and engage in discussion with us when we're talking about, in each disease setting, with each product, et cetera, what's a reasonable threshold for harm and how much information will we have at any given time point. And so I think those are really complex discussions that are, so I would encourage sponsors to come in and engage with us on that for specific products.
Great. David?
You're going down the line?
Yeah, we are.
I don't like the wait answer either, and I think pre-specification of when and how to use interim OS is so important, because I don't want it introduced if it's not useful. If it's just confounding or weak enough that we shouldn't draw conclusions from it, then it doesn't belong in the discussion at the time of accelerated approval, at the time accelerated approval is sought. Leave it out for fear that we err on either side and continue to gather data. If a drug can be approved based on surrogate endpoints and the OS just mixes everything up, then don't use it.
Great. Lisa?
Yes, of course, pre-specification and thinking about the information at each time, coming to talk with the FDA and really planning for what are these decisions being made during your trial. At each time there is a decision to be made, and what's the information you're using, and how is it going to be used? How are you defining harm? And this information going into the decision is important, and I'm sure a lot of sponsors do that, but I think discussing that with the FDA early is what's important.
Great.
Okay, for me, I think a key takeaway really is the need to really continue with the industry, regulatory, academic collaborations here to probably develop tools that are going to help with these quantitative decision frameworks through the clinical trial simulation and predictive modeling.
Thank you. Okay, well, we'll take questions. Please come to the mics if you don't mind, and feel free to call out a specific panel member in your question. Otherwise, I and they will attempt to triage them. And if you don't mind introducing yourself when you ask your question.
Two FDA people right off the bat. Yeah. Wow, this must be an important topic. This is for Jeff, actually, and I think I was really struck and appreciated your point that one of the design features that we have to do is to get 100% post-progression ascertainment of survival events. It's really challenging if we do, as you suggest, we don't wait, we approve, and we look for a subsequent OS readout to make ourselves feel better that it's not going hard the wrong way. If we have a lot of missing data, we just get more uncertainty. But there's one other data element that I was going to ask you how hard it is to get. And we know in situations like multiple myeloma or anywhere where there's a sequence of effective therapies, that one of the ways OS can go the wrong way is inability to get to the effective subsequent therapies because of toxicity, even grade 2 toxicity perhaps. How good are we, or how hard is it in a multi-regional trial to get a good read on post-progression therapies on both arms, not just the second therapy for multiple myeloma, let's say, but even maybe 3 therapies out? And I know that can be complicated, but I just wanted to hear your perspective on what you've seen operationally.
My perspective is biased as a lung cancer doc, where the second therapy, docetaxel, is no one's favorite. So I struggle to think of the second-line therapy that is so disease-modifying that you really have to make sure they get to it, because most of those disease-modifying therapies are being given first. But I'm with you. If that situation exists where there really is a disease-modifying second-line therapy, then I guess that needs to be pre-specified. The fact that patients progressing on a RET inhibitor don't want the chemo is a representation of how satisfying and disease-modifying that therapy, in fact, is in the eye of the patient. So I hate to put it back on you, but that requires a little more thinking about what is that exact situation, and you just got to nail it out earlier, I think.
Not have asked a lung cancer doctor, I apologize. But I'm thinking about multiple myeloma, where the subsequent therapies are, in fact, quite effective, and there's a lot of them. And that's not the only disease space anymore in that setting. So, but I do think that's one of the data elements we should do a better job with.
Yeah.
Hi there. Thanks for the great discussion today. Jeff, you had a number of examples of discordance where PFS, I guess, overestimates survival benefit in your presentation. I'm curious to get your thoughts on the opposite type of error where PFS underestimates survival benefit. Dr. Pazder's group at FDA had recently published in the Lancet Oncology an analysis on a number of lung cancer trials in the immune checkpoint inhibitor setting. In their conclusion talked about drug developers potentially missing opportunities to get beneficial treatments to market by relying too heavily on these early endpoints. Curious to get your thoughts on, I guess, the other side of the coin there.
I agree it's a dilemma. It's a little bit of a different, it's not kind of what we're talking about today, right? Where to some extent I'm thinking about the urgency of therapies that are clearly improved and the OS is opaque. You can read my papers about how much I don't believe in PFS or the issues in the development of this historically. I agree with you. There's got to be better tools we can use. Many of them are just statistical tools and how you analyze that PFS data. It's probably a deep dive for another time. Apologies.
Kim?
Hi, Kim Rathmel, NCI. This has been a very provocative panel, and I have a million questions, but I'm going to ask one that's fairly focused, but I think this is the right people to kind of chew on it. I was triggered a little bit, Jeff, when you said, we need to make sure that investigators are encouraged to track for the long haul. And as we try to think about how more and more people are our investigators and getting access into places that are outside the academic centers, that's a burden that is really almost insurmountable. And yet we have tools, AI and real-world data and other ways. Is there a way to democratize the long-term follow-up of overall survival, or maybe even other endpoints that we could think about. And so the question's for you, but I think, Jeff, what would patients think about this, or, folks at the FDA?
We'll send that right back to Paul for the session that's coming up next, right? But I'll say one thing, the biggest problem in my mind is withdrawn consent. When the patient says, I don't want that extra EKG post-progression, I'm out, I withdraw consent, the site says, okay, no more OS collection, I guess. And it's like, wait. So if we can refine you shouldn't be able to withdraw consent from OS follow-up, right? And so I think those are kind of things that can shift the patient's relationship with the trial. The more we unburden the post-progression period, I think we'll make it more conducive. And so yes, let's do that so that no patients are saying, I don't want the rest of this trial anymore. I'm aware of a trial that expected lung cancer patients to get pregnancy testing for 6 months following progression on first-line therapy. Right, that kind of trial, now that was not a US stipulation, but that kind of burden is not going to be conducive to enthusiastic post-progression participation. So it's a good idea.
Yep, thanks.
Nicole,
any other ideas?
Oh, yeah,
No, I think that's perfect. Yeah.
Great. Okay. Well, the real-world evidence piece I think we are going to come to, which is a really important piece informing this conversation. Bruce?
So Bruce Johnson recently from Dana-Farber. I had a question for Jeff and Tony, and that is that and Jeff, the example you used was the RET rearranged lung cancer. And that population is relatively rare, and you don't always pick it up before they get their initial therapy. And sometimes they're making decisions about dosing based on the first 30 to 60 days, and we may overshoot it, which has impacts on how long they can take the drug. You want to talk about how many patients you need to accurately define a dose that you know that a person's going to be able to take it for years, which is commonly the situation in targeted treatments?
There's a lot in that question. I think to some extent, I forget who raised, the longer a patient does in the therapy, the more we observe them to be doing well without toxicity, the more confidence we have they're going to get on the other therapies that can benefit from, and that ties into dose. Right, and if our drugs are all dosed the right way, this is thinking about a different regulatory conversation, right? If our drugs are all dosed the right way with the right risk-benefit, then we won't have this problem of unnecessary sticky toxicity. What's the right number of patients or the right expertise needed, right? How many RET mutant patients do you need to see in your clinic to be able to get the regimen right? All very difficult questions, Bruce. Thank you for raising them. I'm not sure they're all going to be sorted before a first-line trial, a randomized trial kicks off. And so we may just need a space to say some of this stuff will get sorted in the pragmatic world of post-approval studies. I'm going to avoid. Tony, any other additional thoughts?
I don't think there's much more to add, but yeah, it's a step that you're going to continue have to monitor. Yeah.
Let's squeeze in one more question. Apologies, we'll have to cut it off at that point.
Thank you very much. Mahin Khatami. I retired from NCI. I have 4 decades of immunology and molecular biology experiences, and my discoveries were done at the University of Pennsylvania relating to first evidence for direct link between inflammation and tumorigenesis. My comment, concern, and question are all together that how many more decades are we dilly-dallying in reductionist approaches to cancer? We are saying that we used to say cancer is 100 diseases, and then we said 200 diseases, and the last previous NCI director said that cancer is 1,000 diseases. That's not true. Cancer is only one disease, and severe dysfunction of immune neuroplasticity that we should really pay a little bit more attention to it. Cancer is considered as a wound that does not heal, as it was told many years ago by Dvorak. And I consider cancer is an imbalance between tumorigenesis and tumoricidal side of effective immunity. It looks like we have put our conscience in deep freeze by just going after the business of cancer, not to do something more effective for cancer patients. If we go in that direction, I'm hoping by the new administration that we don't have to spend trillions of dollars, 30 to 50 trillions of dollars has been spent ever since NCI or NIH was established, and we are still nowhere, whether it's this or that molecule or this inflammatory mediator or that inflammatory mediator, that's not the answer. And I have had the answer, but I've been intellectually pushed aside because we do not want to stop the cancer promotion. We are promoting cancer. And cancer war, as any war, I believe, the first thing, the first casualty in any war, including cancer, is the truth. We are not telling the truth.
Okay.
Thank you.
We'll take that as a comment and concern as you introduced it. Thank you very much. Jeff, I think, is going to send us off for coffee. We're all dangerously low in caffeine levels.
Dangerously low. Thank you all very much. We are going to take a brief 15-minute break, and recognizing that there is a large virtual contingency today, we will do our best to observe that time and be back here. Maybe we'll make it a 13-minute break and be back here at 11:50, please. 55.
If everyone could please take your seats, we're going to be getting started with panel 2. If everyone could please take their seats, thank you. Thank you, everyone. We're going to get started now.
Okay, thank you. Wow, that was very orderly. Thank you. I think we're going to go ahead and get started now with session 2 for today, and I want to thank the members of our next working group who have spent the last several months delineating opportunities to incorporate pragmatic elements into clinical studies. We collectively recognize, and has been noted several times this morning already, that clinical trials can be particularly burdensome and may not always provide the results that are generalizable to the patient population that may ultimately use the drug or provide all of the relevant information to optimally deliver the drug in clinical practice. In these instances, additional post-market studies may be requested, and this next panel will focus on this context and discuss their thoughts on identifying and incorporating pragmatic elements into post-market studies. To start things off, I'd like to introduce Dr. Paul Klutz, the Deputy Center Director of FDA's Oncology Center of Excellence. Paul, thank you.
Thank you very much. Thanks, Jeff, and thanks, friends, for having me. I think this is an important panel, as we heard from the commissioner, and I think several other discussants saying, well, a lot can be learned in the post-marketing setting, and how can we do that? So my talk is really kicking off how we look at instilling pragmatic elements into commercial clinical trials and NCI-sponsored trials. So I think everyone knows the issue. The issue is that our explanatory RCTs have been complex and are only getting more complex. And that creates burden and expense to patients, investigators, and sites. But I think, as we all know, evidence is going to still be needed in the post-marketing setting more and more on what the appropriate sequence is, on comparative effectiveness, on comparative tolerability. And in order to get this high-quality evidence, I don't think we're going to have the ability to create a very complex RCT to answer all these questions. So we need to modernize how we generate evidence. And when I think about what it is that we can do to modernize evidence, it's 3 buckets that I can think of that I've seen. One is, even if in the most complex explanatory trial, we can be more efficient and more patient-centric. So things like master protocols, things like statistical analysis plans that make sense, and decentralized trial elements. So get trial conduct out more to where patients live. And we saw that in COVID, and I think we're starting to see that more and more. But not only can you decentralize and make more efficient complex trials, you can make trials less complex, and that's largely what we'll be talking about in this panel. So can you have a focused objective in the post-marketing setting where you can reduce collection of some of the assessments that we do in our explanatory trials? And it is not a fixed price menu. It is a la carte. There's elements of pragmatism you can instill in almost every context. And then the last bucket is something that we've all been talking about. Can we leverage electronic health records? Can we leverage digital health to try to pull information more into our prospective research in an easier way? And I think there's a lot ongoing in that. But for this panel, we're really going to be talking about pragmatic and decentralized elements. So this is the PRECIS 2 framework, and this is how the panel is going to begin to talk about these pragmatic elements. This is a continuum of various dimensions of clinical trial conduct that can be from the most explanatory type, which is restrictive and lots of follow-up, to the most sort of routine standard of care and pragmatic approach. And so they sort of rank it from 1 to 5. And as you can see, and I'll pause on this for just a moment, it goes from eligibility to how the trials are conducted and where they're conducted, to what sorts of assessments and follow-up are going going to be conducted to what the primary endpoint analysis plan is. And when I first saw this pre-cis wheel, I naturally went and thought about all of the clinical trial protocols that I've seen at the FDA over the last 10 or 15 years, and a lot of them are in parts of the protocol that I think we can do a lot of work on. We know Friends, as well as ASCO and FDA, have worked on eligibility criteria now for almost 10 years, if you look at the first paper, and we've done a lot with that pragmatic element. So we've been thinking about pragmatism, and we didn't even know it. So I think every trial can look at eligibility criteria. I think decentralizing some of the elements of trials, in other words, moving the setting more out to where patients live, is definitely an opportunity in many trials. And I think, as you'll see as we talk about this in the panel, as well as some of the ongoing trials, the number of assessments and type of assessments can actually be reduced. And that could be safety assessments being reduced to labs, to even resist PFS assessments, which is, very surprising. We love our PFS, but in some cases, if you're getting OS, you may not need to do that. And that's very burdensome to sites, to sponsors, especially when you look at blinded independent review. And finally, endpoints. The primary endpoint of a clinical trial, being overall survival, is itself pragmatic and lends itself to the ability to do some of these other pragmatic elements as well. But we're also interested in exploring what I would call real-world intermediate endpoints. You've seen a lot of this in the published literature. What can we pull out of routine clinical care that might approach a progression-free survival type of endpoint? Things like time to treatment discontinuation, time to next treatment. There's a lot going on in that space. I think we will continue to learn about those. I wanted to blow up the settings component of the pre-cis dimensions just to say that even that has elements to it that you can select and choose depending on your trial context. So even as simple as e-consent is moving some of the conduct of the trial out to where patients live, and most trials can consider that. And obviously, as you go around the wheel, you can see they get more and more complex from an operational standpoint, all the way to delivering drug to the patient. So I really want to make it clear that I think every trial conduct can consider elements of pragmatism and elements of decentralization in its design. All trials, even the most complicated and early trials, can look to expand eligibility and can look to decentralize some of the conduct. And then the appropriate degree of how pragmatic or how decentralized is obviously going to be context dependent and based on what's known about the drug in the context. So some contexts can be very pragmatic, and this is a slide that I adapted with permission from Janelle Gray, who's done a lot of work on the Pragmatica lung trial, and she, in presenting an update of this trial, went over the protocol and noted that there are no protocol-required CT scans or disease assessments, which is very unique for a registrational trial, and therefore no resist reads, no blinded independent review. Of course, patients are getting their CT scans through standard routine care. There are no specimen collections. There are no protocol-required lab tests, and only unexpected reportable grade 3 or higher AEs are going to be reported for safety. So this is as stripped down of a trial, as pragmatic of a trial as I have ever seen for a trial that has registration or labeling intent. And as Janelle shows, the impact of that on our documents alone is staggering. We're talking about a third of the protocol pages, eligibility criteria cut down to a fourth, et cetera. So it can really have an impact. And we will learn a lot from this trial. So I think why change? I tried to articulate why I think we have many questions and all of them cannot be answered in a highly explanatory randomized clinical trial. I think what are the incentives? They're different from each of the different stakeholders, but certainly patients will benefit from less burdensome trials. I think investigators and study staff who are really overburdened right now will benefit from more simple, less complex trials, more consistent with routine care. I think decision makers like FDA and health technology assessment agencies will benefit from more generalizable evidence that will look more like the US population and those who are likely to take the drug in the post-marketing setting. And I do think sponsors have incentives. They will benefit from more patient-centric trials with faster enrollment and less attrition. That is the goal. And I don't think we can overemphasize how important patient centricity is in this current competitive environment. There are multiple trials going on in the same indication with very similar drugs. And I would think a patient would want to go on a trial, all things being equal, that was less burdensome for them to participate in. So the panel is going to go over a couple of objectives. What we tried to do is create a couple of case studies so that we could dig into some context-specific questions. The first is about characterizing safety and efficacy in an underrepresented subgroup, in this case, racial and ethnic group that wasn't well represented in the pivotal trial. Case study 2 is looking at a safety question, further characterizing the risk of accumulative toxicity and potential mitigation strategies. And case study 3 is not just characterizing the safety and risks, but actually providing substantial evidence of efficacy and safety in a biomarker-selected population where you want to expand the indication beyond what the approved biomarker cutoff was. Essentially, you want to broaden an indication. So in conclusion, I want to stress that it is not all or nothing. There's no such thing as a pragmatic trial or a non-pragmatic trial, a decentralized trial or a non-decentralized trial. There are elements of both that should be considered and can be integrated in most trials. It is context dependent, even in trying to create cases, as the panelists next will show you. There was a lot of it depends. It depends on the safety of the drug, it depends on what we know, it depends on a lot of things. And so you should come to the division with your ideas, and we'll look at the context. I think it's important to understand and appreciate the sponsor's concerns about uncertainty, costs, and risks. Anytime you do something different, you're putting a lot on the line, and I don't think we should look the other way to that. But what I want to say is I do think that the uncertainty, the costs, and the risks will decrease with experience. And I know that will be the case over time. We have several trials that are ongoing, Pragmatic along just being one of them, but I know there's some cooperative group trials that are looking at instilling pragmatism in some of our other questions that we'll learn from and hopefully de-risk the future and make evidence generation a little bit less burdensome to all involved. So with that, I welcome the panel, and I think this is going to be a great discussion. Certainly the commissioner is very interested in this, and as I think we all are, so thank you.
Okay, thank you, Paul. I'm Rich Shilsky. I'm the former chief medical officer at ASCO, emeritus professor at the University of Chicago, currently serving as the PI of the ASCO TAPER study, which by design incorporates many pragmatic elements. Although it's not a registrational trial, not a randomized trial, but we've learned a lot through that study about how well pragmatic elements can work in making a trial much more accessible and making the data much more generalizable. I want to invite my panel members, and I'll introduce them as they come up. Alan Chen is the Global Clinical Head, Oncology Late Development at AstraZeneca. And come on up, people. Tricia Camacho, VP, Head of Clinical Research Collaborations and Medical Evidence Generation at Bristol Myers Squibb. Ava May, Patient Representative. Neil Maripol, who's Vice President of Clinical Research in Oncology for Flatiron Health. Peter O'Dwyer, co-chair of the ECOG Akron Cancer Research Group and Professor of Medicine at the University of Pennsylvania. And Donna Rivera, Associate Director for Pharmacoepidemiology in the Oncology Center of Excellence at FDA. So I think we should jump right in. I just want to pick up on a couple of points we already heard about. So obviously the commissioner has charged us very clearly to think about how we can be more effective in generating evidence in the post-market setting, where he and I think probably we perceive that there's a lot of unanswered questions that need to be answered in a way which is formalized enough to get reliable information to inform clinical practice or inform subsequent regulatory decisions. Paul, I think, made the point very clearly that which I strongly agree with, that we should get away from using the term pragmatic clinical trials. This is not an all or none. Trials are not pragmatic, or they are or they're not pragmatic. Any trial can have pragmatic elements incorporated in it, and we as a community of trialists need to look more closely at every element around that Prey-C2 wheel to think about when we're designing a trial, how can we make it more pragmatic? Because generally speaking, we recognize that there are many potential benefits to introducing pragmatism into our clinical trials in terms of making them more accessible, making them easier on sites, incorporating a more generalizable patient population, generating data that is more broadly applicable to the intent to treat population, and so on. But there are also some risks of incorporating pragmatic elements. We're going to try to bring those out in this panel discussion. And one way potentially of mitigating some of those risks is to, at least initially, focus introducing more pragmatic elements in the post-market setting. So with that, I'm going to have a seat, and we're going to start the discussion. I'm going to maybe start with Donna at the end there to talk a little bit about, from your perspective, why in fact did this panel decide to focus initially on the post-market setting for more pragmatism?
Sure, thanks, Rich. So, I think when we think about what the objective of a pragmatic elements that are introduced in a clinical trial is, it's really on reducing burden, really most importantly for patients, but on the whole ecosystem. And I think you heard the commissioner say that while there is an opportunity to think about pragmatic element introduction in the pre-market setting, in the post-market setting, you have a situation where there's a well-understood safety profile, and safety is paramount. You have well-understood and available efficacy data. And that means there's an evidentiary foundation to think about how we might introduce pragmatic elements. Because pragmatic elements introduce, depending on the element, different types of risks, different types of uncertainty. And really, one of the reasons to start with the post-market setting is exactly that, risk reduction. We think about pragmatic elements as a continuum. So the flexibilities that are introduced, they're really dependent on the trial context, and most trials can introduce some level of pragmatism in the conduct, in the measurement, or in the eligibility. For example, it's not collecting no safety data or all safety data, but maybe selective safety data reporting. So I think the question we're really trying to ask, and the reason that this group thought a lot about it and started with post-market, is thinking about asking the question not yes or no, is it a pragmatic trial? Rather, what pragmatic elements can be included in the trial, and how do we make a trial fit for purpose?
I'm glad you used that term, because I remember that was one of Janet Woodcock's favorite terms about talking about fit for purpose data collection. And of course, it's linked directly to what the trial objectives are and the intended use of the trial data. So Alan, let me turn to you and ask, maybe from the sponsor perspective, what do you see as the potential benefits and maybe some of the risks of going more pragmatic, particularly in the post-market setting?
Yeah, thanks for the question. I think, as you said, the main consideration from an industry perspective is what is the goal or the objective of the post-market study. If the goal is to mainly generate data to inform the broader clinical practice, then I think there's the opportunity to introduce more pragmatic elements, because in that sense, there's potentially less risk to the sponsor. On the other end of the spectrum, if you're trying to generate data that potentially could lead to a label expansion or some kind of label update, then it's likely that you'll need to have more rigorous data collection that would be sufficient to support regulatory decision making. And that would be key to have some early discussions with regulators to get a sense of what is the bar for potential regulatory decisions. And then somewhere in the middle, if it's a study that is not necessarily intended to lead to any kind of label update, but maybe lead to updates in guidelines such as the NCCN, then maybe you're somewhere in the middle where you want to generate data that would be sufficient to get some kind of recommendation in the guidelines, and there's sort of a middle ground where you can introduce some pragmatic elements, but sufficient to inform the guidelines people.
Yeah, that that leads to a question maybe that I'll ask Ava to comment on, because as Commissioner Califf pointed out, when the FDA approves a drug, they approve a drug for a specific use in a specific population. But of course, we know that there are many other questions that often remain unanswered about how best to use that drug, questions that are very important to patients and clinicians. And so Ava, what do you think about this whole area of introducing more pragmatism into clinical trials, in particular in the post-market setting, but maybe over time to carry over even into the pre-market setting?
Well, I really like the idea of starting with the post-marketing setting for this case, because I think it gives us an opportunity to really build on the patient partnership, because it's a setting now we know that the drug is safe for a specific use, but there are a lot of unanswered questions, and in this case particularly, there were under-recruited, very important racial and ethnic groups that need to be part of the clinical research in order to really know that the drug is as effective and is safe. So this gives us a chance to improve the patient experience of participating in the clinical trials by reducing the burden and by making it as streamlined and as patient-centered as possible. But it also gives us the opportunity to do implementation strategy testing, because we're still in a research environment, and so we can really look at how to improve uptake and awareness and interest in the treatment, or not, really, based on the patient experience. And that's what makes me very excited about this.
Yeah. Neil, did you have a comment? Or you look like, Neil, you look like you're about to say, okay, great, we'll come back to you. We'll come back to you for your non-comment. So this is great. I mean, I think, I think the big issue that this panel was grappling with over the last several months is, the decision on when and how to introduce pragmatic elements really comes down to a benefit-risk assessment, like everything else in drug development. And there are benefits and there are risks, and the risks can be mitigated by introducing pragmatic elements initially in the post-market setting, and that's the reason for this focus. I want to get into now some of the scenarios that Paul introduced, because in order to sort of exemplify some of the issues and some of the risk-benefit considerations, we worked around these three scenarios. One has to do with studying a population that may have been underrepresented in the initial registration trial, whether that's defined by racial ethnic characteristics or by geographic location, or by other measures. Age, for example, we know older people are frequently not well represented in clinical trials, even though they are well represented in the intended use population, and so on. So if we focus initially on that scenario, there's been a lot of work that's been done, Paul alluded to, by ASCO and Friends of Cancer Research collaboratively with FDA on recommending how to broaden eligibility criteria for clinical trials, recommending how to simplify clinical trial operations. But Alan, maybe you could talk a little bit about the extent to which some of those recommendations are being implemented, what industry may see as the benefits and risks of doing so.
Sure. So in the context of a post-marketing study looking to generate additional safety and efficacy data in an underrepresented subgroup, such as African American patients, that's an area where I think pragmatic elements can help to expedite the completion of that type of study. So, for instance, you mentioned eligibility criteria. Rather than using the standard eligibility criteria that we would use for a pivotal study, which can be very restrictive, maybe having looser or relaxed eligibility criteria that would enable you to find those less common patients in your studies. Obviously, whenever you introduce broader eligibility criteria, that potentially creates more heterogeneity in the population. So that's a potential risk, is that by introducing a broader population in that respect, that potentially the treatment effect is diluted, right, with a broader population, or perhaps the safety looks different because you've introduced patients maybe who are a bit less fit than what you had in the pivotal trial. So that's sort of a risk that needs to be considered. But I think overall, depending on the goal of the study, that type of risk might be acceptable.
Yeah, I mean, that's clearly the trade-off. And, obviously, those patients who were not incorporated or not included in the pivotal trial are still going to be treated in the real world. And in a sense, wouldn't it be better to have the data on the efficacy and safety in those individuals than not to have it at all, so that we can better inform their treatment going forward? So, there's been a lot of discussion. Paul brought up decentralized trials. There's been a lot of discussion about making trials more accessible in communities, in community practices where most patients are receiving their care. Again, there are opportunities and some risks associated with doing that. I want to turn to Neil because obviously Flatiron has been operating a large community-based practice network, doing research in that, providing some technology services that you might want to talk about to facilitate clinical trial participation at that level. So why don't you talk to us about that a bit.
Thanks, Rich. When thinking about representativeness in clinical trials, the gold standard in a way would be to actually conduct the clinical trial where patients receive their care. And that is probably the strongest way to achieve true representativeness. So because most cancer patients get their care in community settings, de facto, you would like to conduct clinical trials in those settings. And there are lots of examples of community networks, community practices participating in clinical trials research. The national cooperative groups are one setting. There are other consortia. The idea of introducing pragmatic elements is one mechanism to improve accessibility that you referenced, Rich, for community sites. By increasing the feasibility of participating, because there are pragmatic elements that make the conduct of the clinical trial less burdensome on sites, for example, the quantity of data collection required, and also increasing the attractiveness to those sites, because the attractiveness of a clinical trial, both to patients that are seen in the community versus an academic research center, and to the clinicians in those settings, may have some nuanced differences that one has to think about in terms of study design for a study that you might want to conduct in a community setting. In addition to the pragmatic elements related to study design, there are also operational enhancements, if you will, that are enabled by technology that can improve both the efficiency as well as the feasibility of studies and reduce site burden, such as the use of telehealth, telemedicine, for remote visits. This has become routine part of clinical trial activity, which is particularly important in community settings. The identification of patients using automated means, machine learning, and other patient matching technologies at the point of care to reduce the burden of patient ascertainment while also ensuring that all eligible patients are surfaced to the clinician investigator at the point of care. And then finally, technology is now gaining traction for the automated transfer of electronic health record data directly into clinical trial databases, as well as electronic data capture systems through a point-and-click technologies that again reduce site burden in terms of the data transfer into study databases, and also have the potential for reducing errors because you're not dealing with the transcription from one source of data to a data capture system for the clinical trial.
Great, thanks. So, another element of pragmatism that's been widely discussed and in fact recommended in many cases has been reducing the amount of safety data collection in clinical trials for drugs that are already marketed, where generally speaking we have a pretty good sense of the safety profile of the drug, and as long as the intended use population is not radically different from the drug in which the population in which the safety data was originally generated, it's not likely that we're going to learn a whole lot more by continuing to collect reams and reams of safety data, particularly low-grade toxicities. And this is an area that NCI is moving into now, and with some recent recommendations from a group I think that you co-chaired, Neil. But I'm going to ask Peter to comment a little bit on reduced safety data collection in clinical trials. And Peter, also, I might ask you to talk just for a minute about what I consider to be a bit of a myth, which is that community sites don't participate much in cancer clinical trials. NCI has a 50-year history, I think, of doing community-based clinical trials through the CCOP and now the NCORE program, and you're very much engaged in that now. So maybe first talk a little bit about what you see as the benefits and potential risks of reduced safety data collection, and then maybe talk a bit about how robust is the community network, and are they really ready for this level of engagement?
Yeah, thanks, Rich. And in terms of the issue of developing safety data in the post-marketing setting, excuse me, I'd go back to one of Ava's comments just now, is that the structures that we're talking about today have the potential to allow oncologists to better understand how to administer the drugs and how to handle the side effects. And the scenario that we're developing should be thought of not as an end product in itself, but as a learning environment in which we will figure out which pragmatic elements in what scenarios can be best used. I hearken back to how did we do it before we had this context of pragmatism? Well, we just figured it out. Oncologists communicated with each other, and they talked about side effects, they talked about management strategies, and so on. But trials to do this were not that common. The potential of this is to advance it into a research setting. When I talk about the research setting for a real-world community oncologist-led set of research objectives, I think about the minority and underserved NCORP sites that are in our group and in all of the cooperative groups. And what they tell me in many cases, particularly those that involve safety net hospitals, is they simply can't do the complicated trials, the drug company sponsored trials. They don't have the ability to complete all of the intermediate endpoints, all of the scans, all of the MRIs and so on, and sometimes specialized testing. They can't do this. So trials like this immediately can expand the footprint of clinical trials. I'm a bit concerned, however, that we have a robust set of community sites that participate in our studies. But many of them recruit very few patients per year. And it's not a financial thing, it's usually a bandwidth thing, and it's a resource thing. So the advantage of this pragmatic approach and pragmatic designs in simple trials are that we can increase the number of oncologists participating in clinical research. And this is really important because that's how you reach real communities, that's how you achieve penetration into rural areas and into other underserved areas in our cities as well.
Great, thanks. Tricia, I want to turn to you. I recall during some of our working group meetings, you made a comment about how pharma typically collects lots and lots of data, but not all of it is used or useful to the FDA in making regulatory decisions. Certainly not all of it ultimately informs or appears in the drug label. So, as we try to move more toward pragmatic elements in clinical trials, which may result in less data collection, how do you think about how to engage with the agency in deciding what data collection you can forego when setting up a trial?
Sure. No, thank you for the question, Richard. And I think it's a very passionate topic for me. I think when we think about and why we focused on this post-marketing setting, that is an opportunity that's ripe for application of the pragmatic design. And even an opportunity that after we get a label indication for a subset of populations, either as monotherapy in combination, do we take the opportunity as sponsors, and that sponsorship could be on the manufacturer, and it could also be in the context of the academic center, the community center, the consortia, to look back at the data sets that we've collected aligned with statistical analysis plan and the objective of the study and say, was all of this applicable for the objective of the trial? And I think on the sponsorship side, we also look very much at trying to ensure we have a grave responsibility to ensure we have a full understanding of the risk-benefit profile. So the safety profile, full characterization of that, and of course AE is a special interest, and of course the efficacy profile. So you don't want to miss out on collecting data. So we hold ourselves to high accountability to collect sufficient data, and then oftentimes more than sufficient data, to ensure that we are prepared for any questions that the agency may have. But I think at the inflection point, it happens on the other side of that, when we can then, after achieving a label, an indication, look back and say, and work with the agency to say, are there things that we can shift now looking forward at the lifecycle management of a drug or protocols in the post-marketing sense that we may no longer need to collect what we collected previously? And I think that's an opportunity for us to reflect on.
Yeah, thanks. So I'm going to stay on you for another minute as we move to our next sort of scenario that we discussed, which is about the not infrequent need to further characterize a specific toxicity that may appear in a registration trial. And the specific example we chose for this discussion is a chronic toxicity, one that may be delayed in appearing, one that may be protracted, and particularly a neurotoxicity of some sort. And I guess the issue is, what are the opportunities in that setting to introduce more pragmatic data collection or more pragmatic elements into a clinical trial when you have a specific goal of better characterizing a specific toxicity? And maybe talk to us a little bit about how you would approach that.
Yeah, no, I think when we thought about the case examples, we thought mostly about themes, right? As opposed to asset themes or target themes. We thought about the themes of the pragmatism of the study, so the diversity piece, and now, of course, the safety follow-up. And this could be encompassing anything. To your point, a cumulative safety event or some type of event that you could anticipate that could influence the risk-benefit profile, the longer-term risk-benefit profile. So how do we do this on studies that we may already be able to have a long-term follow-up for survival, just to the earlier discussion that we had with the first panel? But there could be also situations where it does further protract the study for safety events, and how do we do that? So I do think, again, it goes back down to the identification of what is the specific AE of interest. Can we work with the agencies to ensure that we are collecting that at the appropriate cadence? And I think it was also mentioned in the prior panel, which it resonated with me, which is when we understand when patients drop off study, right? I think when you consent a patient, this is under a contract of they would continue to participate in the study. As that study potentially gets further protracted, there is some fatigue that the patient could go through as well as the site. And what we want to do is try to understand how we can get over that fatigue or help ensure that we don't lose that data over time. And so I do think that's a critical component that I think is helpful too.
So presumably, if we're trying to better characterize a safety signal, that might include learning about things like, who's at risk of developing that specific toxicity? What is its onset? What is its duration? What strategies might be applicable to mitigate that toxicity? And I guess I'll ask Peter maybe to talk to us a little bit about, what kinds of more pragmatic assessments might be feasible, while at the same time trying to zero in on a very specific type of safety signal.
Yeah, this is an easy one, of course. Neurotoxicity is the example that we showed, that we talk about in the paper. It's a good example because it's cumulative, number one, and it can be long-term. And strategies to mitigate it are really not available. And why are they not available? Because we've never had a test that can reliably predict the depth of neurotoxicity and whether it gets better. It's an entirely subjective type of analysis. So clearly, when something is subjective like this and qualitative, bringing it out into the largest group of oncology specialists that you can find is much more likely to get you a way of dealing with it. And so I think that while we describe a study that involves the description of the toxicity, its severity, and its recovery, it would be even more ideal that a study like this should also be able to evaluate ways of dealing with the toxicity, ways of improving the patient's condition. And certainly, if one envisages large numbers of patients on studies like this, as one hopes there will be, this would provide a mechanism of doing it. But it was emphasized before by Alan that one of the risks of having an adaptive approach to studies like this is heterogeneity in the type of treatment that patients get, which may affect the interpretation of the efficacy signal. So that type of error needs to be controlled for always.
Yeah, thanks, Peter. And I just want to interject one comment here, because people often ask about, in circumstances like this scenario, couldn't the sponsor just collect real-world data to better define the safety signal? And again, like everything else we're talking about, I think it depends a lot on the context. And in part, it depends on what's in the drug label and what is part of routine clinical practice for using the drug. So for example, if the label or the clinical practice guidelines or whatever don't specify a particular way of ascertaining the toxicity, of managing the toxicity, and so on, it's not likely that real-world data is going to reflect any of that, because it does not happening in the real world. So the real-world data is only as good as what doctors do with real-world practice, and to the extent that doctors are guided by clinical practice guidelines or drug labels, maybe real-world data can be helpful, but I suspect that oftentimes it will not be, in ascertaining a specific kind of toxicity. But one thing that can be important, I think, is getting more input from patients about these types of toxicities and how they tolerate them. And Ava, I'll turn to you maybe to talk a little bit about how digital health technologies might be informative here, or how hearing more directly from patients about their experience with the treatment can be valuable.
Thank you. Well, I think it's really important always to remember that not every patient will want to play the digital game. And so for the patients that are willing to embrace digital technologies, there's so many opportunities, I think, that are much more efficient and much more patient-centered, starting with the electronic consent form. That if a relationship is begun with that, then it's very easy to follow up and develop patient relationships. In a lot of ways, just making patients know that they're valued, from things like thank you notes, reminders about appointments, thanking people for going to appointments, thanking people for completing surveys. All of that brings about a feeling that the patient has that they are an important part of a trial. Then when it gets to reports and surveys, the patient-reported outcomes, of course, are very important, and talking about a much more streamlined series of surveys where it's really the important serious events, but also the opportunity to identify events that may not be that serious, but that are impacting people's quality of life, that may have been overlooked in the accelerated approval process, but may emerge as real issues that are manageable, that, as you were talking about the qualitative oncology discussions, same with the qualitative advocacy group discussions. We talk about things that a lot of people feel, but we don't know if it's just a few of us or it's really a system. So I think that these are great opportunities throughout the complete research project that can improve the patient voice and the robustness of the patient voice.
Great. Thanks. I want to move us along because we're going to get a little bit short on time. So our last trial objective, or last trial scenario that we discussed was expanding an indication or looking for a new use. And the specific example we discussed was for a biomarker-directed therapy, expanding the definition of the biomarker-eligible population by changing the cutoff, changing some other aspect, using a different assay, maybe switching in some cases from a biomarker defined by IHC to a biomarker defined by a genomic criterion, FISH or gene amplification. All of these things are happening routinely. They all have some risks and benefits associated with them. Alan, let me turn to you to kick us off in this discussion about if we're going to consider doing this sort of thing, what are some of the considerations we need to keep in mind?
Sure. Yeah, and I think, again, this boils down to whether the goal from a sponsor perspective is to try to seek a label expansion, right, to broaden the label to get a different or broader population than what was originally approved. And if that is the case, then I think a lot of attention needs to be paid to the test used to identify the new population, right? If it's already using the same approved, validated biomarker test, then maybe we can rely on local testing, and there's no need for the additional burden of submitting a sample for central testing and waiting for the result. That's always a factor in getting patients onto studies. But if it is a brand new biomarker test that hasn't been used to support an approval before, then likely we won't be able to be pragmatic in that regard, and there will be a need to provide tissue and central testing and all of that. So I think that's a discussion point that would be needed to have with the FDA in advance of starting a trial like that, to see if there's room for acceptability of local testing for the biomarker. And obviously, if this is a study, an additional study, there's already been a large pivotal study done and the safety profile is already pretty well understood, then maybe there's an opportunity there for, as we were saying before, to simplify the AE data collection, right, in these studies and make it less burdensome to the patients. So I think there's some elements that can probably be pragmatized in a way, so to speak.
Yeah, so in this scenario, the key element really seems to be central versus local testing, and how feasible that is. And I guess I'll turn to Donna maybe to talk a little bit about that from the FDA perspective.
Sure. So I think Alan outlined it nicely, that when we're thinking about this, you're thinking about when you're answering your question, how are you making this fit for purpose, right? How's a trial fit for purpose? And I think in this example, there are some inherent risks, and those are really around the data heterogeneity and the maintenance of data integrity. So the focus in this specific instance, when you're thinking about what's important in this example is how we understand maintenance of efficacy, something Peter mentioned earlier, and really how we identify the biomarker selected population. And so the precision of that, the potential of introduction of variability in the local versus central testing. And if you're trying to understand a very small differential efficacy change, if you're trying to understand it among biomarker measures, the data may not be easily interpretable. I think the question is what is the magnitude of benefit you're expecting, how you think about introducing these elements, and what the interpretability of the data is going to be. And then if you layer on additional pragmatic elements, is that furthermore, for example, differential assessment frequency, are those layers going to increase risk? But I do think that there are benefits, and those benefits could be in a more representative trial, in an opportunity to include rarer populations, faster completion, more access, as has been mentioned. So I think it's really such an individual evaluation of how the question you're asking fits with the design, and can you create evidence that is maybe more generalizable to fill a knowledge gap.
Yeah, it seems to me that this is a case where really understanding the biomarker performance is critical, because if you just take the example of moving a cutoff to a sort of lower level so that you include more patients in the study. Well, if that new segment of the patient population doesn't actually benefit from the treatment, then two bad things could happen. One is, depending upon the proportion of the overall trial that they represent, you could either dilute the overall efficacy signal for the entire trial, or you could end up concluding that the entire population benefits when all the benefit is still driven by the people who were at the higher end of the biomarker range. So this is really going to be a critical consideration, I think, going forward. In the interest of time, I'm going to move us sort of right to the last couple of minutes here. And maybe, as Keith did in the first panel, just go down the panel and ask each person for any final reflections, and particularly with respect to, what do you see as incentives to move the field more toward pragmatism? It's been reported that, despite the fact that a lot of people have been talking about this for a long time, the number of trials that actually include a lot of pragmatic elements is still pretty limited. So there's certainly some concern or resistance about moving in that direction. But I want to give each person sort of a last word to reflect on what we've been talking about. So Alan, maybe I'll start with you and we'll just go down the line.
Sure. So I think, from a sponsor perspective, obviously the goal, the best sort of scenario or outcome for us would be a label enhancement or label update for a broader population or a different population. But even bar that, there's also potential benefit for sponsors in, as I mentioned at the beginning, speaking to guidelines groups like the NCCN to see if guidelines can be updated to potentially make some kind of recommendation. It may not be the same level of evidence, but at least some mention if there is data generated in a different population that could support broader access, as long as you're able to generate data that there is a positive benefit risk in that group. So for example, since I work at AstraZeneca, Tagrisso is primarily indicated for patients with the common EGFR mutations, but there are data showing that in some of the less common mutations, there is efficacy there. And so the NCCN guidelines do actually have a recommendation for these less common EGFR mutations that both osimertinib and afatinib can be used in that setting. So that leads to reimbursement in the U.S., which benefits sponsors. So I think if you're able to generate data, even if it doesn't meet the bar for a label update, there is potentially other criteria or other outcomes with guidelines updates that can be beneficial for sponsors.
Right, Tricia?
Yeah, no, I think sponsors, I think there's a myriad of advantages that we have and incentives. I think first and foremost, there's a social contract to make sure that we have diverse populations. Having to do a subsequent study, to do that, I think it can be costly. So trying to find the efficiencies in the primary study are going to be incredibly important. I think also trying to bring in novice trialists and trial sites. I think we have a tendency to go to where we have experience and comfort and our incredible trial performance, but we also want to make sure that that has the broader applicability, and so I think we have a huge incentive there. And then to the earlier point around longer-term follow-up, dropout, I think we do want to ensure that, one, we find that right balance of addressing or doing our adequate job of answering the scientific question at hand, and at the same time not overburdening the entire medical community in terms of that population of patients, care providers, investigators, and of course sponsors. So I know we have limited time, but I'll transition it to
Ava.
So two other things. I think that the sponsors can really help the sites with recruitment and engagement of a lot of times a new patient population by offering culturally competent communications pieces and other tools to help them with the engagement and the recruitment. And I also think that sponsors, when possible, need to remember that there are other burdens to patients for participating in trials, such as stipends for transportation, child care, parent care, everything else, and to try to look at other ways to minimize the burden besides just what happens within the trial.
Thanks. Peter? Neil, sorry.
Yeah, we've heard today again and again evidence that all stakeholders in this world are really seeking clinical trial innovations and ways to streamline the conduct of clinical trials. At the same time, we're in this really heavily regulated, risk-averse environment where folks are weighing the desire for innovation and moving the needle against the risks of doing so. So this is not going to be a flip the switch, the world is going to change tomorrow. But nothing is going to move the needle like success. And so my plea is that we try collectively, meet early and often with the agency to discuss potential application of pragmatic elements, and that we publicize and highlight successes. The FDA in June, I think, put out a review of all of the submissions that involved real-world evidence. I could see something like that related to pragmatic clinical studies, where these are the elements that were implemented in this submission, and this was the outcome of that evaluation. So let's be transparent. Let's publicize successes as well as failures and learn from them, and let's move the needle.
Great. Peter.
30-second version. We understand from these discussions that pragmatic designs have benefits for patients, for the investigational sites, and for the sponsors. And most of these relate to the burden on the patients and the cost throughout the system. We have to recognize we live in a resource-limited environment. This can allow us, the incorporation of these elements can allow us to learn more about different treatments faster and across the community. The role of these trials is evolving, and I hearken back to Paul's talk that, that we need to learn how best to apply these, where to apply them, and so forth. And I am very confident that, that this is a big step forward in the philosophy of how we apply clinical trials to the cancer problem.
And the last word, as usual, goes to the FDA. Anna.
Thanks. Well, I will try to tie the threads of the panel and say I think I have a generally optimistic view, and I think, as you heard from the commissioner, it's a problem that we have to solve. And so I want to conclude with two thoughts. One is talking about risk mitigation. I think there are very many types of risk in drug development, and part of the process is assessing that risk, evaluating it. This panel is here because everybody from their own perspective is evaluating that risk. And some tools I think that can be helpful are, of course, coming to talk with the relevant review divisions, but also special protocol assessments that provide an opportunity to talk about the design up front as a request of the IND. We have 2 new guidances on point-of-care trials and decentralized trials that really outlay important aspects of design. And interestingly, in Project 5 and 5, we received over 30 submissions across various cancer types. And one important factor, I think, was that not all these questions had registrational intent. So registrational intent is just one part of this picture. It's bigger than that, which leads me to my second point, which is that I think pragmatic evidence generation is a shared responsibility. And this shared responsibility is across regulators, sponsors, payers, providers to create more patient-centric evidence generation. And we've talked a lot about incentives. Leading up to this, I did an exercise to look at the benefit-burden ratio of all the PRECIS criteria among sponsors, providers, and patients. And in almost every category, it was beneficial to the patients. So to me, improving patient outcomes is an incentive, and I think we have the opportunity, the catalytic opportunity with everyone here to try to bring trials to patients in their communities. And that said, we recognize there are challenges, and we hope over time, with examples, we can reduce the potential risks. So to me, the question is how, not when or if, but how with limited resources, so many questions, so many evidentiary needs, we can bring elements of pragmatism to clinical trials and move the field forward together.
Great, thank you. So we are the only thing standing between you and lunch. We have, I'm now told, 4 minutes for questions. So if you have a question, please come to a microphone and identify yourself. We'll try to take at least a couple of questions. Yes.
Hi, everybody. Dave Fabrizio, Foundation Medicine. I have a comment/question on the first consideration that you guys laid out in the executive summary around the underrepresentation populations. And the clear focus is to make sure that the clinical trial population represents the intent to treat population. But from a biomarker perspective, and if a trial is incorporating a biomarker design, if that biomarker was trained on the wrong population, that problem will only magnify. So I'm curious to get the panel's perspective on how you think about also a biomarker perspective on making sure underrepresented populations are part of that biomarker definition.
Yeah. Anybody want to bite on that? Sounds like the whole problem with AI in a nutshell, right? Depends on what you train on. No comment. You stumped the panel.
That's our only question.
I know there's 3 people standing there. We're going to take one more question. And I think you were the next one up, so please go ahead.
I'm Padma Rajagopal from the NCI. I'm a breast medical oncologist and data scientist. So I appreciate the prior question in front of me, and also wanted to point out not only the problem of representation from sort of a limited data set as part of your problem with real-world use, but also the problem of imputation. If you are extrapolating some of your data that you're missing from limited data sets, that's also going to be a problem for real-world data applications. So just wanted to add that to what the panel can chew on. Thank you very much.
Okay, any response to that? Any comment?
Maybe I would just say that, I think what you're suggesting as far as imputation, as far as real-world data, what we're building on, it so much depends on the context of the question and what's being asked and how much data you're imputing, what was the a priori statistical analysis plan. So I think certainly a lot of considerations, and I think I agree with Rich's earlier comment that in some cases real-world data will find it very challenging to answer those questions, but certainly could be a complementary strategy to the addition, and of course the way in which we may use real-world data in the context of routine clinical care and the whole objective of really embracing pragmatic elements in a learning healthcare system is one hopefully we're all working toward.
All right, I think we're going to conclude there. I want to thank the panel. I also want to thank Brittany McKelvey from the Friends staff for really helping coordinate all of our work over the last several months. So thank you, Brittany. And I will turn it back to Jeff.
Thank you. Thanks very much for the great discussion. Hopefully it can continue over lunch since we had to cut it a little bit short. But you can find lunch now being served out in the foyer. There should be a number of stations. Please feel free to bring your lunch back in here, utilize the tables here. We'll break for about 30 minutes before we'll have the chance to hear from NCI Director Dr. Rathmel. So please be back in the room at 1:20 promptly for that session. Thank you.
If everyone could please take your seats, we'll be getting started in 1 minute. Thank you.
Okay, I think we will go ahead and get started with our afternoon sessions. Please feel free to continue eating, though. We'll keep this hopefully a nice casual conversation. But I'm super excited to have Dr. Kim Rathmell join us for today's discussion. As I'm sure many of you know, that Dr. Rathmell is a renowned kidney cancer expert and has been an influential leader in cancer research and patient care, from treating patients with kidney cancer to conducting research that has advanced the underlying science of kidney cancer, and participated throughout her career in a number of large-scale important projects that hopefully we'll hear about today, both in her pre-NCI days and working with the NCI throughout her earlier portions of her career. But while Dr. Rathmell is the 17th NCI director, the way that timing has fallen, I think it's been almost about a year, but this is our first time that we've been able to extend an invitation as the NCI director to join us at the meeting today. So we're really looking forward to hearing from her. Hopefully we'll have a chance to learn a little bit about some of her recent experiences running the NCI, but then thinking about the future of cancer research as well. So would love to just start with some of your perspectives and hear about going back, I suppose 12 months in some instances, perhaps it feels like 12 seconds, perhaps in others it feels more in the magnitude of multiple years. But would love to hear a little bit about what some of your early impressions were when you transitioned into the NCI, and what were those expectations and how did they materialize as you transitioned into the role?
Yeah, no, that's great. It's great to be at this almost year mark to kind of reflect on those kinds of questions. I've known the NCI for a long time as an investigator and other ways that I interacted with the NCI, so I thought I knew the NCI. It's even bigger than I ever thought that it was. I found the thing that I learned almost immediately and have only learned more and more about every day is how much the mission really drives the output there. The way people, staff scientists, everyone is committed and motivated and knows exactly where it is that we need to go. That's a great kind of organization to get to lead, because it's very easy to navigate, because people are so focused on one very unifying mission. So I've just enjoyed every minute of this time. So, and just like you said, it feels like it's been a flash, and it feels like I've been there for a long time. It's felt very comfortable. And
is that shared mission something that surprised you most, or were there other aspects that were totally unanticipated?
So I think that probably surprised me most, other than little surprises of being in the government and learning how to navigate all of that. I think the pieces that I've enjoyed maybe most, just to expand that a little bit, is being able to be a part of different parts of the whole research enterprise that have really not been in my immediate wheelhouse before. You know, we're really spending a lot of time, like this has been a great conference for thinking about access to care and how we deliver care to underserved communities and to rural America, and how you get clinical research in all of these spaces. That's been a major effort of the last year. One that was a part of my repertoire, you know, came from Vanderbilt with, you know, a lot of rural area around, but not in the how do we get research into these spaces. And so that's just one example, and it's just tremendous to get to play in this really large space.
Right. Well, hopefully we'll come back to some of the implementation side of things, but given the audience and the focus of many of the topics discussed today, particularly on collaboration and the future of drug development, I wanted to ask you about, I know you have to leave after this session, but the following session today is related to developing a coordinated approach toward validating the use of circulating tumor DNA as a potential endpoint. And I'm not going to ask you any specifics on that particular topic per se, but when you think about some of the large-scale potential that the NCI has, in larger scale partnerships like endpoint validation, how do you see the NCI playing a role in leading those types of collaborative efforts?
Well, I think we play a leading role from everything from, you know, funding the R&D research that gets to these technologies. I think one of the interesting challenges and opportunities is how many different technologies make up these liquid biopsy, cell-free, ctDNA assays. But, you know, I think we do very well at creating consortia. So we have a large collaborative that works together to make sure that we're connecting the dots on these emerging technologies and where they can have a big impact. But we also are proactively including them in, for example, some of our bigger clinical trials, the follow-on trials from the MATCH study, like Combo Match and Mylo Match. They both will be collecting liquid tumor biopsy so that we can move from where we are today, really focusing on tumor-specific biomarkers into, you know, something that's much more readily accessible in the blood. So we can't just wait for that to happen. We have to be prepared and ready. So those kinds of various different ways are really vital. I think the other thing that we're doing that I'm really excited about is, you know, looking at the way we use these kinds of biomarkers for detection, these multi-cancer detection tests. You know, they are out and in use, but we really still don't know exactly who and how and when to use this information and what we can learn from it. I think we will learn some of how we think about cancer in a different way, but we can't assume that, and we can't just believe it. We have to prove and gather the data to do that. So the Cancer Screening Research Network, which was just launched, will be one vehicle that we'll use to learn how we use all of these various tools. Yeah. So I'm so glad that it's being discussed here, and it's going to be on the front of everyone's minds for a while.
Yes. Well, I certainly want to thank you and your colleagues at NCI for your willingness to partner with all of our external organizations, but it's been a driving force, and we're certainly grateful of the expertise. With that in mind, in terms of facilitating collaboration, and even in the cancer screening space, you know, that's been an important part as FDA is starting to think about the evidence that needs to be generated for not only those types of tests, but in clinical studies. Can you talk a little bit about how you see the NCI and the FDA working together?
Yeah, absolutely. Actually, that would have been a good answer to the first question too, about, you know, what was one of the things that surprised me most was, you know, how close the collaboration is with the FDA, and how, I mean, we saw it already here in the first morning sessions, is how very forward-thinking folks at the FDA are, and how willing to be, you know, sort of a part of, you know, designing the clinical trials that will make a really big difference for people. So we've been, you know, across the NCI in various different iterations, meeting with the FDA regularly to think about the pragmatic clinical trial elements. And this is, not new, but it's new that it's on everyone's sort of tip of their tongue, and where we can start to think about it for not, I really loved the session before about it's not pragmatic trials, but pragmatic elements that we bring to our clinical trials.
Yeah.
So,
do you see unique opportunities sticking on the pragmatic elements concept? Is this becoming a necessity for some particularly cooperative group studies that are particularly resource limited at times?
well, so first of all, so I'm a Midwesterner. I come from Iowa, so pragmatic just feels like it should just be state common sense. Let's do common sense things, and that so that common sense makes sense when we're applying clinical trials that are testing important questions that might be beyond the registration of the drug, but to how we actually use those drugs. The NCTN is a great example. There are formalized partnerships with NCTN leadership, NCI, and FDA to sit down and look at clinical trials and be able to partner as effectively as we can. I think to your question, is that essential? I think it absolutely is essential. As we look to democratizing clinical trials, distributing them into much broader spaces, and partnering as broadly as we can to get clinical research, I think, as a part of standard of care. Yeah. This is not, clinical research is only after we've exhausted all of the known therapies. There isn't a cancer out there where our treatment is perfect. There's more to do in every different venue. And so being able to have that opportunity, we probably didn't have the infrastructure before. We don't quite have it now, so we definitely didn't have it before. But we didn't have the ideas and the drugs and the opportunities before, but we have that today. So it's tremendously exciting to think about how we use the whole infrastructure of cancer care delivery and as a part of how we think about doing clinical research and getting new studies out. And I also really loved the comment that I'm just going to echo from Peter O'Dwyer about that this is not just how we learn what the next best way to treat is, but how we learn how we do that and how we use this as a learning healthcare system. It's really, really exciting and essential.
And going back to some of the comments that you made around expanding research access and equity, can you talk a little bit about the role that the NCTN is playing that, particularly with the NCORE network?
Yeah, in particular, the minority and underserved NCORE sites, they are really the mainstay of having clinical research reach underserved communities. But this happens in other places as well. And I would say this is a priority across our cancer centers. This is a priority across our NCTN sites as well, because I like the National Cancer Plan because it's a very great way to ground what is the work that we need to do. How do we have the biggest impact? Look at one of the goals is to eliminate inequities. If we eliminate inequities, then we reach all of the target population of people who have cancer or risk for these cancers. If we limit our knowledge to only those who have access today to academic centers or to restricted networks, we miss out. So we miss out on bringing these discoveries as far and as wide as we want them to get, but we miss out on the data as well. And we don't learn as much as we could from having a much more diverse setting of population there.
In the session that preceded the break, there was discussion around the need to support non-traditional sites or non-academic sites as part of a component of improving access, generalizability of the data that's being generated in clinical studies. Do you see opportunities that the relationship between NCI and the biopharmaceutical sector could be expanded beyond what it even is now?
Yeah, I think this is a time where we all have to work together. And so it's NCI, it's biopharmaceuticals, it's everyone who has a vested interest here. One example that I think I'll share is to look at some of the innovations, for example, that the VA is doing. So that it's another area that we're partnering so much more than I had really anticipated until I got into this role. So the VA has this unique healthcare system that covers the entire country, uses a lot of telehealth, right? And really prioritizes using telehealth and distributed care as well. And so we've been working with them just to bring more infrastructure for clinical trials so that NCTN studies, for example, can be opened through VA sites. But we can learn a lot from them as well and be able to think more pragmatically about, what are we imposing as barriers, for example, to a telehealth opportunity in a clinical trial. That's something that they just do every day and is upfront when they're thinking about, can I open this trial? It has to involve telehealth often. And so that can help us think about whether a trial could be conducted with a remote patient interaction. So just as one example, I think we all have to learn from each other.
In instigating some of those things, are there like parameters or guidelines or things that the larger NCI could suggest to some of the sites in order to make that more of a at least frequent consideration in implementing trials?
Yeah, so this is exactly why we have these kinds of conversations. And this is the when we say the NCI, right? It's the partnership with NCTNs, and it's working with the cancer centers as well, and thinking about where we can put those. And I would say, and the NIH. This is not cancer specific. This is generalizable to medicine and research overall. But we can learn really from all of them.
And could you touch a little bit along those lines in terms of your interactions with other centers at NIH? Obviously, there's things that may be done related to cancer in other institutes as well. Is that something that that you anticipated you'd like to see more of moving forward?
Yeah, well, I always knew that there was a lot of other. As a medical oncologist, cancer researcher, my funding was always NCI, but I did kidney cancer, so I always wondered about NIDDK. Now I'm an NIDDK researcher because NCI directors can't have a laboratory in their institute. And it turns out there's a whole interest group there of kidney malignancy investigators. And so I've been able to plop right into that group. It's really, really fantastic. So I think the world has gotten very intertwined in the kinds of issues that we're all grappling with. How to deal with big data, right? How to manage AI in the research that we're doing. How to think about biorepositories. These kinds of bread and butter fundamental things. How we train people to become scientists and physician scientists. These are cross-cutting. We all work on these things. You asked about research particular to cancer. I think, first of all, there is very clear cancer research that happens in many other centers and is funded by the other institutes for very good reasons. But there are also lots of things that are happening that are, at first might not appear to be cancer, but are going to be so relevant to what we do. I mean, we look at the the discoveries that come, in areas of, metabolism, and they find their way back to being, really important for cancer. So it's, it's a whole spectrum of gray that's in there. And
You mentioned this buzzword a little bit, so I'm going to hang on to it. But the phrase AI in healthcare sort of has, applied to every imaginable aspect of of healthcare, and end to end, really. But how do you realistically see AI providing the greatest short-term benefits?
So this is why we have a deputy for data science. So Warren Kibbe just joined us, and it's been wonderful to have his expertise, but there's lots of good experts. So that's a really hard question to answer. I love when I'm seeing with AI emerging in how we manage imaging. We just had a big set of publications around the Human Tumor Atlas Network, the HTAN, which in order to analyze this incredibly complex data, looking at three-dimensional intersections of multiple tumor types, we can't do that with our normal patterns of logic. We need AI to help to do that. So that it created tools to allow us to really understand the complexity and depth that's in these tumors. Fantastic. As well as just practical tools for how we assess images, how we manage some of the kinds of things that need to be done that require a workforce. We need to augment our workforce with AI because we just simply don't have enough people sometimes to do all of the work in managing patients or managing data. But just even in how we interact with the data that's coming, it's just phenomenal. I've been plugging a tool, and I'll plug it again here, called Nancy, N-A-N-C-I, currently only available on iPhone, but soon for Android users as well. That was created, this was from our training branch. It was created by trainees, for trainees, and uses AI to help interact with our publications, and allows you to very quickly navigate through publications and see where some of the details are that you might have not be able to see without reading the entire publication, and be able to predict what other things you might want to read. It's really just a fairly granular tool that can enable, I'm reading a lot of new literature that is very new to me. I don't have time to read all of the papers in every single topic area in cancer. And I read a lot of papers, but tools like that can help us to find the high priorities and get to them and summarize them very succinctly.
When you think of some of these uses of AI and identifying potentially patients that may be eligible for different clinical trials, do you think the technology will sort of solve itself, or do we need to be thinking about alternative sort of regulatory kind of validation strategies, for lack of a better term? And it's interesting. I ask because you talk with policymakers, and there is also a polarizing view of the role of AI in healthcare. One, it's sort of this panacea, and on the other, it may take over the world inappropriately. So how do you sort of find the middle ground, and will these things just resolve themselves through more experience and research?
So yes and yes, I think. Some of this just it will still be to be determined. We're still learning, and we're still seeing, and some of the things that we learn will lead to the guidelines and the practice patterns and ways that AI fits in and is commonly used. So some of that's going to be organic, but it can't all be organic. I think we have to have smart people who understand what is happening behind the scenes, and who are a step ahead from those of us who aren't computational biologists or data scientists, who can be a part of those discussions so that we get to the right place at the right time. Plus people who are ethicists and people who can provide some of those guidance. I think we can't move at a snail's pace, but it has to be just a little bit of both in there. The idea of using AI, that's why I asked that question. Could we use real data tools to just pull this information forward? Because I think there are so many other parts of our society and parts of our world that are utilizing data to help inform decision making, and healthcare has, for good reason, been a little bit slower to adopt. Data privacy is a really important component. How we do this safely and with confidence and don't erode trust from people absolutely is essential. But we also will have to move forward.
Yeah.
So.
Now that you've had this sort of vantage point from the epicenter of oncology research and care, if you had the ability to kind of address one short-term obstacle that you think will be really critical to the future, where would you start?
Ooh. I only get one?
Well, you can,
yeah,
you can have a couple, but.
Yeah, well, I mean, I'll go to the low-hanging fruit, which is, the funding for, government-funded research is not, does not fit the size that we are today, or the expectation of what the American public has for us to accomplish. So that's a real, that really keeps us from expanding some of our clinical trial infrastructure, for example, in the way that we would want to, or to be able to incentivize some of the research that we want to move forward. Just a lack of funding is a reality of our day-to-day. But I think the other issue is really around it's people, but it's, are we reaching the right people? Are we creating the workforce that can prepare us to move into the next generation? Enough of the right workforce who are going to stay with us and be able to propel us through all of these amazing things that we're all talking about here today, and be able to get that into the future that we're looking for.
Yeah. Of all of the decisions that come across your desk in any given week, how frequently is funding limitations a component of that? All?
Yeah, yeah. Every day.
Yeah. Well, with that in mind, I think some of the collaborations that were described earlier, or the expansion of various public-private partnerships to hopefully begin to work together will ideally offer some opportunities. With that in mind, I think that was one motivation around trying to align stakeholders around sort of the second iteration or the reignition of the cancer moonshot. Inevitably, that will begin to likely take a different form, whatever that may be. But as you think about some of the lessons learned through those discussions, do you see any future benefits that you would really point to around sort of that all-of-government approach to advancing cancer research?
Yeah. I think that the all-of-government approach was actually really pivotal. I mean, I wasn't here before, but the doors feel very open to engagement. I already mentioned some of the exciting things with the VA, things with the FDA. We partner with ARPA-H on things like the data fabric project, because data science is important for literally everyone. So being able to have those kinds of cooperative interactions, I think once that door is open, I can't imagine how it gets shut again. So that's just the way that we are wired now, and the way that that things get done. It doesn't always happen that doors open and people actually walk through and collaborate. The tight funding climate probably helped to bring us all together a little bit more rapidly. I don't want to continue that just to do that, but the relationships of the organizations are really very solid, and so if that is the biggest outcome of this, that's huge, and really will allow us to tackle cancer in ways that just were not possible before.
It's very heartening to hear, and hopefully something that can be a model into the future. When you, sort of with that in mind, perhaps more broadly than outside of, beyond just the halls of government, so to speak, but what do you see for the oncology community? What do you think this community needs to focus on over the next decade to actually make the transformative differences that were sort of envisioned with those moonshot goals?
Well, so I'll bring it back to the National Cancer Plan again. Sure. In order to achieve a decrease in mortality, hitting a number like 50% decrease in 25 years, definitely going to take prevention, early detection, advances in treatment, and optimal care, right? The whole, what I've been calling kind of arc of the patient experience, all of that has to be front and center. We can't just focus on one. We have to hit all of those components. And then the rest of the National Cancer Plan will really take it up. We already talked a lot about hitting underserved communities and eliminating inequities, utilizing data, but the workforce is absolutely vital. And then engaging every person. I mean, that part of the National Cancer Plan is absolutely my favorite, because it's engaging how we talk to people about cancer, how people begin to think about cancer. Because of the advances that we have today, the ways that we can prevent and detect cancers, the way we treat it, people don't have to fear cancer the same way that they did before. And we're already seeing that happen. Helping people realize that that's happening is a big part of how we reach that new future. So I lean into the National Cancer Plan, really find that it's incredibly valuable for thinking about the map for how we go forward.
One more question, not to make things seem overly daunting here, but in addition to sort of that national plan, how much of that is global?
Oh, that's a great point. So we actually make the point that the National Cancer Plan is I mean, it's a fairly flexible and adaptable plan. So it absolutely applies to this. And cancer is not a United States problem. Cancer is a worldwide problem. It's one that we can make more inroads by making partnerships and by learning from others, as well as sharing experience. We know, and this has come up I think a couple of times already here, that as cancer gets more complicated and discrete, we almost need the whole world so that we are able to understand the cancers based on their submolecular phenotyping and on the rare cancers and some of the nuances of cancer. We absolutely need the whole world to work together. And the things that we can learn from them is as much as they can learn from us.
Well, on behalf of our organization and everyone here, let me just thank you for your leadership. You certainly have hit the ground running over the last 12 years. I hope that we'll be running with you for a while and appreciate all that you're doing. And thanks for being with us today.
Yeah, thank you very much. My pleasure.
So I think we will just move directly into our next session this afternoon, which will focus on, as I mentioned, defining a common strategy for using ctDNA as an intermediate endpoint for prospective studies. I think many of you here today probably know that Friends has had an opportunity over the last couple of years to work with a really terrific group of stakeholders and collaborators as a piece of this large research partnerships to explore the role of ctDNA. A goal of this work is to provide data-driven considerations for the use of this measurement in clinical trials, and that's what will be the focus of today's discussion. And this has really, truly been I guess we'll get out of the way before they move us here. This has really been a remarkable effort, in large part led by Friends Director of Regulatory and Research Partnerships, Dr. Hillary Anders, who has done just a marvelous job in keeping things running forward on this project, and she's going to present some of those results today. And thank you, Hillary, and we look forward to the discussion to come, and thank you so much.
Yeah, thanks very much.
Thanks, Jeff. I appreciate everyone being here. This is really exciting. I'm here on behalf of not only our organization. Is there any way you can make the actual slides a little bigger? I don't need the notes as much on the monitor. I'm here on behalf of not only the working group that you'll hear from later today, which is our white paper working group, but also from our working group for the CT Monitor project. In both cases, there's a little bit of overlap, but we have a lot of stakeholders. I think there's 120 folks who are on my monthly call. So we have a lot of support on this project, and I want to thank everyone who has helped us. So today we are focused on ctDNA as an intermediate endpoint in oncology clinical trials. Intermediate endpoints can identify safe and efficacious drugs sooner. Validation is critical to ensure reliability of the intermediate endpoint. There are general requirements for validation, including methodological consistency across studies, correlation with clinically meaningful outcomes, such as overall survival, and robust meta-analyses. Over the past few years, we have some recent examples, including pathological complete response in breast cancer and minimal residual disease, or MRD, in multiple myeloma. ctDNA holds promise as an intermediate endpoint, so that's circulating tumor DNA. Initial data demonstrate that decreases in ctDNA levels from baseline to on treatment associate with overall survival. There are a few key questions that are important in this space, and our CT Monitor project has been helping to answer them, including, do ctDNA dynamics differ by cancer type and treatment modality? What is the optimal timing for ctDNA measurement? How should a molecular response be calculated? And what assays are most suitable to be using in these circumstances? To use ctDNA as an intermediate endpoint in regulatory decision-making, FDA expects aggregate meta-analyses both at the patient and the trial level. The Friends CT Monitor project asked the question, do decreases in ctDNA levels associate with improved overall survival? Our initial pilot project we called Step 1, which was an advanced non-small cell lung cancer treated with an anti-PD-L1, included 5 trials of 200 patients, and this was really truly a pilot to understand whether we can aggregate patient-level data. And what we saw was that a greater than 50% decrease in ctDNA associated with improved overall survival. This showed us that it was feasible to aggregate heterogeneous trials with varying collection time points and assays. We then moved on to Step 2, which included a few different modules, but Module 1 focused on advanced non-small cell lung cancer, this time treated with a tyrosine kinase inhibitor. We had 8 different trials of about 1,000 patients, and here we looked to see whether trends were similar in a different treatment modality. Rather than looking at a % change, we noticed that there were a lot of patients who had non-detection of ctDNA on treatment, and when they went from detection at baseline to non-detection on treatment, we considered that clearance, and those patients who had clearance of ctDNA had improved associations with outcomes compared to those who had persistent detection. This study showed us that different treatment modalities may require distinct definitions of molecular response. For our CT Monitor Module 2 analysis, we focused on a study across 4 RCTs with about 1,000 patients, comparing an anti-PD-L1 with or without chemotherapy to chemotherapy alone. And I'm really excited to share with you our findings from this study for the first time. We had 2 goals to build on previous findings. The first was to assess change in ctDNA and associations with overall survival, and a separate treatment modality, in this case chemotherapy. We were also interested in confirming our results from the anti-PD-L1 data, and also exploring how ctDNA assessed at different collection time points early in treatment impacts interpretation of results. So some of the definitions that we have, this just is an outline of some of the early time points that we're talking about, looking at the various weeks. Our baseline was defined as a ctDNA measurement taken up to 14 days before index. In this case, index is randomization. Our T1 time point was up to 7 weeks from index, and our T2 time point was 7 to 13 weeks from index. And that was based on, as you'll see in the blue and pink shaded boxes, the darker shaded boxes were the first time point for the various trials, and then the lighter shaded boxes were the later time points. For our approach, we defined molecular response as either ctDNA that was non-detected on treatment or had an X% decrease in ctDNA levels. In this case, we looked at 2 different cutoffs using either a 50% or a 90% cutoff. And our non-molecular response category included those with a less than X% decrease in ctDNA levels or an increase in ctDNA levels. For our analyses, we looked to assess associations between change in ctDNA. We defined that as a molecular response and OS at T1 only. And then we assessed the influence of ctDNA timing on interpretations, so molecular response associations with OS at T1 and T2. We compared the T1 results to the T2 results, and then we established what we called confirmation categories using T1 and T2. Here we're starting to take some lessons learned from RESIST. We know that in RESIST you often have more than one measurement to kind of establish what the molecular or the response category is. And so we looked at what patients' response categories were at T1 and T2. So the first analysis was looking at associations with overall survival for anti-PD-L1. On the left, we're looking at a 50% cutoff, and on the right, we're looking at a 90% cutoff. And we see that, as we've previously seen for the anti-PD-L1 treated advanced non-small cell lung cancer, defining molecular response as a greater than 50% or 90% decrease associates with improved overall survival. We then looked at the chemotherapy data. Again, on the left, we're looking at the 50% cutoff, and on the right, the 90% cutoff. And again, for chemotherapy only, molecular response associates with improved overall survival. We do see slightly weaker associations when compared to the anti-PD-L1 data. We then compared the T1 results to the T2 results, and here on the left, I'm showing you the overall survival for anti-PD-L1 using a 50% cutoff at T1, and on the right, that same cutoff, that 50% cutoff for T2. And we have other comparisons that all looked pretty similar to this, and so for all comparisons, both in the anti-PD-L1 data and the chemo data, looking at 50% or 90%, we see similar trends for molecular response associations with outcomes at T1 and T2, but T2 tended to have a stronger association. When we compared the T1 results to the T2 results, we performed likelihood ratio tests that showed that associations at T2 alone provide similar information to using models that assess both T1 and T2, again suggesting that T2 may have stronger associations than T1. For both the anti-PD-L1 group and the chemo-only group, about 80% of the patients fell into the same response category at T1 and T2. And when the response categories did not match, the T2 time point influenced associations with outcomes. So our overall takeaways from the Module 2 analysis are that molecular response associates with improved OS for patients with advanced non-small cell lung cancer treated with anti-PD-L1 and or chemotherapy. The magnitude of change is stronger in the anti-PD-L1 group compared to the chemotherapy alone group. And ctDNA collected up to, at T2, so that 7 to 13 weeks time point, has a stronger association than T1, up to 7 weeks. However, the trend is apparent in both, which I think is really important to consider because we do know that there are other factors to consider besides just the statistical outcomes when looking at T1 and T2. So our next step was how should ctDNA be incorporated into the next wave of clinical trials to enable future meta-analyses that support using ctDNA as an intermediate endpoint. For this, in order to perform aggregate trial-level meta-analyses, we first wanted to align on a standardized, harmonized approach for data collection. And then the idea was that that approach would then be included by sponsors in prospectively designed trials. And then finally, we'd be able to perform trial-level meta-analyses using that aggregated data. So what I'm sharing with you today, we established a working group that wrote a white paper with recommendations. In our recommendations, we focus on advanced non-small cell lung cancer treated with immunotherapy. And our working group included industry sponsors, diagnostics developers, FDA representative, patient advocates, and academics. And we focused on a few areas for alignment, which our panelists will go into more detail. But we looked at assay characteristics, the timing of sample collection, patient inclusion considerations, approaches for calculating a molecular response, and approaches for comparing treatment groups using molecular response, and finally, some considerations for the meta-analytic approach. Our recommendations were first to report data about assays and assay outputs that ensures transparency to support trial poolability. As you'll hear from our panel, there's a lot of things to consider when you're choosing an assay for these trials, and we want to ensure that we're not trying to recommend that everyone's using the exact same assay, but that we understand how the assays were used so that we can determine whether they can be pooled. Use an assay that is sensitive enough to detect ctDNA in at least 70% of patients at baseline. Collect a baseline ctDNA measurement on cycle 1, day 1, preferably before the patient receives treatment, and collect on-treatment measurements for the next 3 cycles, as well as at 6 months. So we have a few outstanding questions after the white paper. We're still trying to understand which assays and assay attributes are most suitable for these trials. We need to better understand how a molecular response should be calculated. Is that a % change? Should we use clearance? When is the best time to measure ctDNA? So we're still kind of looking at windows, but is there an optimal week that we should measure it? And our goal is to answer these questions in future meta-analyses. And for next steps for the CT Monitor project and beyond, we want to encourage sponsors to incorporate the recommendations from the white paper into prospective clinical trials. We've heard from some of our groups that they're already starting the planning process of how this might be done, so we hope that that continues. CT Monitor Module 3, which is our last module of our Step 2 analyses, includes different cancer types and different treatment modalities. And we want to analyze those and then use lessons learned to inform a meta-analytical approach. And finally, establish the meta-analysis plan for combining trials combining data at the trial level and the patient level from these prospective trials. And with that, I want to again thank all of the working group members for all of the work that they did. And at this point, I'll invite Dr. Charu Agarwal, who's a professor of medicine at University of Pennsylvania. She'll be the moderator for our discussion, and we'll also have all of our panelists come up.
Thanks, Hillary. That was an excellent overview. And I would just like to introduce our panelists. I'd like to welcome Zoe Asaf, who's a principal scientist, liquid biopsy technology leader at Genentech. Following her, we have David Fabrizio, who's vice president, partner innovation at Foundation Medicine. We have Leon Frater, who's senior director, global regulatory leader at Johnson & Johnson. We have Anand Pathak, medical officer, Division of Molecular Genetics and Pathology, Office of In Vitro Diagnostics, Center for Devices and Radiological Health at the FDA. We have Carol Vallette, who's a patient advocate, and then Paz, who's acting supervisory associate director, Division of Oncology 2 at Center for Drug Evaluation and Research at the US FDA. We're going to have a lot of things to go through, but we will have time for questions and answers at the end, so please save your questions. I'd like to start off by saying this was a lot of discussions were generated in our working group, and I'd like to highlight the positive results from CT Monitor to date, which can really influence how we manage patients in our clinic. I want to find out, Paz, maybe I'll focus on you. How are these and other emerging data informing the use of ctDNA as an intermediate endpoint when you look at clinical trials, and what other analyses may need to be completed?
Thank you very much. So we saw some interesting data presented by Hillary that in the CT Monitor project that changes in ctDNA were associated with overall survival outcomes in patients with advanced non-small cell lung cancer. And we saw these analyses done at the patient level. And so FDA is very encouraged by these results. We're really interested in bringing effective therapies to patients faster. That being said, we still need more data. And to accomplish that, we oftentimes will look to things like meta-analyses so that we can look at the association between ctDNA and other long-term outcomes, both at the patient level and at the trial level. And so today, I think it's been really great to work with the group here about how do we kind of prospectively think about what data we need to collect in future clinical trials to generate the data that we might need to develop ctDNA as an intermediate endpoint.
Leon?
Yes, so as Hillary already talked about in her presentation, our working group determined and laid out in the white paper what parameters should be harmonized in the future prospective clinical trials to enable a more homogeneous trial-level meta-analysis. So these recommendations included a collection of blood samples, which we recommended at least 5. These should include baseline and 6 months post-treatment initiation. Also, what's important is minimum requirements for analytical assay validation. For those, we recommend that they should be consistent with the set of protocols proposed by BloodPAC. And also, we should ensure a sensitive enough approach to detect ctDNA in at least 70% of patients at baseline. Finally, approaches to defining and measuring ctDNA response to enable comparison of clinical benefit in one group over the other.
Yeah, I think these are so important, right? I think incorporating these in clinical trials moving forward will require a little bit more work, but we need to probably homogenize the way that we approach it, including sample collection. I want to focus a little bit on timing of ctDNA collection. And Zoe, maybe I'll come to you first. And one of the most critical alignment topics that we had discussed during our working group was the timing of ctDNA measurements, because once these trials start, we can't really go back and try and collect more samples. We've seen that T1 and T2 can both relate to overall survival, but over and over again, we're seeing that perhaps T2 or the 7 to 13 week time point may be more critical. We are recommending that we get a baseline sample as well as measurement along the time point during the first 3 cycles. Can you elaborate why we talked about these timings?
Yeah, thank you for the question. So from the CT monitor analysis, like we saw from Hillary's presentation, we saw a really nice association between survival-based endpoints and on-treatment changes in ctDNA at those early time points. Saying that we didn't want to pigeonhole sponsors into really specific timings. It has to be 6 weeks, 9 weeks, 12 weeks, because we acknowledge there's some variability around how studies may give treatment and do also radiographic imaging. And so instead we anchored to the treatment cycle. So if you do baseline, cycle 2 day 1, cycle 3 day 1, cycle 4 day 1, like we recommend in our white paper, then that should hopefully allow for some flexibility in the clinical trial design while also enabling pooling of the data, which is the point at which we're really excited to get to. So we hope this provides some flexibility.
Carol, can you provide a patient perspective? It's so important to hear from you about the feasibility of such an approach.
Yeah, sure. Well, that's why I think it's important to look at it from the burden or the incentive patients will have to be in this study. We know people need to go to a clinic for their treatments, and if we can align the ctDNA collection, even though it's a simple blood test, with that time, I think we have a much better chance to have people enroll and stay with the program. I live in a rural state. Sometimes people need to drive for several hours to go for their treatment. They need to have someone drive them. There may be childcare issues, time away from work, all of that. And just to carve out more time for yet another test, if it's not on that cycle, would be burdensome to folks. So I think the more we can align and make it less of a barrier for people, for patients, the more we can enroll folks in the trial and keep them in there.
And it's so important to be able to correlate this with radiographic responses. Paz, what are your thoughts about that?
Exactly. I agree with everything that Carol just mentioned. And to add on to that, we do have an interest in collecting ctDNA around the same time as RECIST or imaging assessments per RECIST. We want to better understand the relationship between ctDNA endpoints and radiographic assessments. We want to know what the lead time might be with ctDNA versus radiographic assessments. And so we need this data to better understand that. So for example, if you're getting a ctDNA drawn a month ahead of a radiographic assessment, we want to know, is that positive ctDNA result, is that because the ctDNA assay is more sensitive than an imaging study, or is it because it was done a month ahead of the imaging assessment? So we need to gather that information to better understand that relationship.
Yeah, and hopefully in the future we'll be able to come in with a ctDNA assessment ahead of radiographic response and potentially design trials where we can act upon ctDNA information. Carol, you were going to say something.
The other thing I just wanted to add is that occasionally patients do need to take a little holiday from whatever they might have been scheduled for their medication for whatever reason, blood levels are not right, whatever. And again, this flexibility would allow for that. So people can still continue on with the project, with the trial.
We also recommended in the white paper a 6-month time point. And Leon, could you elaborate on why this was chosen and how we got there?
Sure, of course. We acknowledge that the best approach to assess durability of response would be by measuring ctDNA at every cycle for the entire trial and perhaps compare it to IR RECIST progression or clinical progression. However, this may not be feasible. PFS6, on the other hand, is a durable clinical response that we know, which can be assessed in 6 months. Koch et al. Reported in JAMA in 2020 that systematic review and meta-analysis using data from 60 published immunotherapy randomized clinical trials in advanced solid cancers found that 6 months PFS in immunotherapy arms estimated 12 months overall survival very well. So therefore, based on that, we recommended ctDNA sample to be taken 6 months post-treatment initiation to assess whether there is an association between ctDNA and PFS6.
Yeah, I think durability of clinical response is something that we've seen time and over again, especially with ctDNA correlations, not just with response rate, but with the 6-month time point. Paz, can you talk about an FDA perspective? How would this be viewed across trials and in the future?
Yeah, absolutely. We definitely have an interest in how long we are expected to have a response to therapy. And it would certainly strengthen ctDNA endpoints if we're able to use ctDNA to assess durability of response. If we draw parallels from radiographic assessments and overall response rate, we do look at things like Kaplan-Meier estimates of overall response rate. Sometimes we do look also at what percentage of responders might have a response for 6 months or 12 months or even longer. And so these things are of interest for ctDNA endpoints as well. So we'll certainly be looking to see whether or not these ctDNA assessments at 6 months, if that potentially also correlates with long-term outcomes, and that's something that we could look at in a future meta-analysis.
I'll come to you, Dave, next about, I think the big question out there is really, is there a specific assay that should be used? Or maybe there isn't. Walk us through this.
Sure. So first of all, I'm up here representing a diagnostic perspective, obviously not the diagnostic perspective, but we've sort of been down this road before of thinking about how to think about guidelines for big complex challenges, big complex sort of assays. And the one thing we didn't want to do is force ourselves into one particular type of technology. We have to be very cognizant of the fact that there exist many different ways to calculate tumor fraction in the blood. And those ways are at different iterations in their evolution of technology advancement. And so we definitely don't want to stymie that advancement. And so rather than rely on a specific technology, we relied on a set of attributes, which is perhaps the sharpest sword that we have to wield, right, to ensure consistency of data collection and application of the technology. So if you think about how technologies validate their performance, focusing in on that, I think, gives us a good way of approaching this issue.
We talked a lot about, once these trials start reading out, there'll be different technologies. Are there certain attributes that you can define today, maybe perhaps, that we should look at for the future?
Yeah, happy to kick this part off. From a diagnostic assay perspective, there are core assay principles, I think, that are included in our recommendations, and a lot of it goes back to the blood pack recommendations. But looking at things like limit of detection and limit of blank, your assay sensitivity and specificity, and your reproducibility. There are other core aspects of a technology for monitoring ctDNA that we have to think about, such as the impact of clonal hematopoiesis, right? And so this is a clear need for assays to be able to account for how they are filtering this out. So CH signal can give you the sense that there might be tumor fraction in the blood when in fact those are somatic variants not derived from the tumor cells. And so whether you are taking a hard approach by sequencing white blood cells and filtering those out by understanding what exists there, or you're taking an algorithmic approach, we need to be transparent about the performance of those two different solutions, but also make sure that we allow the flexibility of those two solutions to evolve with the technology itself.
Zoe, can you talk about this? Even in clinical practice right now, we have so many different diagnostic assays that are available. Most of them are reporting tumor fractions, some are reporting CHIP reported associated mutations, some are not. We are looking into the future and seeing methylation panels emerge. Can you talk about how these variables may affect the heterogeneity of the data that will be available, and what are some key specific findings that we should really align on?
Yeah, thanks for the question. I completely agree with everything that Dave just said, and just to elaborate a little bit more on the chip issue. So we know that there's these contaminant signals in our ctDNA results from basically biological false positives from chip mutations as well as private germline variants. And one of the fundamental differences in the assays out there is how they deal with that. So there's the tumor-informed approach, the non-tumor-informed approach, the algorithmic correction, the PBMC clearing. And again, echoing that transparency, one of the recommendations we incorporated into the white paper is to ask that technology providers summarize their per variant and per sample false positive rate due to these PBMC or white blood cell-derived alterations. And we really hope that that kind of approach will help with the poolability of the data at a later point. The other thing we're seeing right now, because the space is moving so quickly, is there's a lot of heterogeneity in the types of ctDNA results you can receive. You can get variant allele frequencies, you can get tumor fractions, you can get tumor molecules per ml plasma. And so everyone has their favorite metric, and we're not saying you shouldn't use those, but please, at minimum, take a look at one of the tables in our white paper that specify the minimum reportables per sample. And again, this should get us to a point where at a later date we want to do this meta-analysis. We actually have poolable data.
Such an important point. And Leon, you had mentioned earlier about sensitivity of the assays. You know, apart from all of the attributes, we should be using assays that are able to detect mutations or ctDNA in most of the people. Can you walk us through some of these recommendations around the LOD and the VAF, and then I'll come to Anand after that.
Sure, absolutely. So one of those is that we should be, the assay should be sensitive enough so that they can detect ctDNA in at least 70% of the patients. And the rationale for that is, as we laid out in the paper, is based on the literature, approximately 70 to 85% of patients with advanced non-small cell lung cancer have detected baseline ctDNAs when using an assay with an LOD of approximately 0.1% VAF. So therefore, we recommend that assay should be sensitive enough to measure a majority of the patients with detectable levels of ctDNA. The other one is the LOD of at least 0.3% VAF. And again, the rationale for that is that based on the CT Monitor's findings, in patients with non-small cell lung cancer with anti-PD-1 treatment, this data demonstrated that when using an assay with LOD of as low as 0.3% VAF, a 50% and a 90% decrease in ctDNA is associated with improved overall survival.
Anand, from an FDA perspective, could you talk about specific assay considerations across the board?
Yeah, first I would like to say that and acknowledge that we currently do not have any FDA authorized tests for detecting ctDNA changes for any purpose. If CDER is considering using this biomarker as an endpoint, some considerations with regard to the assay that detect the biomarker include how the test detecting the biomarker was designed. Tests each have their own unique design with their own cutoffs, and they may actually identify different patient populations. In general, key considerations when evaluating the data generated by multiple tests is the poolability of the data. Assays with similar properties in terms of analytical sensitivity and clinical cutoffs, along with other parameters such as the false positivity rate, may help support the poolability of assays. Limit of blank in particular is an important attribute to consider for these assays as well. For as if your limit of blank is too close to the clinical cutoff, that would actually lead to a high false positivity rate. Also, the precision and imprecision of the assay at the analyte levels analyzed may also be helpful to understand. For the delta ctDNA changes being considered here, the delta changes may be unlikely to be very meaningful at levels below the LOD 95. This is because at levels below the LOD95, the positivity for a variant that is really present becomes stochastic and thus may become an unreliable call. Also, the device cutoff for clearance and the differences in these cutoffs between assays is important to consider. Data from this current study has shown that not detected at baseline and not detected on a treatment time point or clearance from detected at baseline actually may be meaningful in terms of association with outcomes. Also, as David and Zoe have pointed out, there's evidence from studies that have been presented at meetings and published in the literature that for ctDNA measurements to be reliable, germline and chip variants are removed.
So far we've talked about importance of ctDNA, using it as a clinical endpoint. We talked a little bit about timing and considerations for patients, as well as attributes of the specific assays that need to be reported. I think one of the key things for discussion is also how do you define molecular response? You know, you've collected all of this data. What are the time points, firstly, to use and define the molecular response? And what other considerations may be there using advanced non-small cell lung cancer patients treated with immunotherapy as an example? Zoe?
Thanks. So yeah, in terms of what to consider with molecular response, one thing I think is worth pointing out is you really do need at minimum 2 time points. We get a really common question of, oh, can I just use 1 time point? In the literature, it shows it's really great for patient risk stratification, but what we're trying to do here is develop an earlier intermediate endpoint that will pick up on treatment-specific differences in survival, and so you really need to look at on-treatment changes in ctDNA levels, and that can be your intermediate endpoint. The other consideration is that the prevalence of your molecular response category is going to depend on the metric that you use. So we've seen in the CT monitor work, for example, that ctDNA clearance, at least with the technologies and in the settings we've looked at, isn't super prevalent. And so it can lead to you being unable to perform the analysis if you have too few patients in that molecular response group. So in that case, we recommend looking at something like percent change and find that 90% is a useful cutoff, at least for risk stratification. Saying that, because the technology is changing so rapidly, it could be that a different cutoff or a different analysis approach is going to be even better. And so again, it's really important to start collecting the data now in a harmonized way so that we can do that meta-analysis at a later point.
And as more variants get reported, these calculations may become challenging in the future. I think that's why it's so important to have variant level information being reported on these trials. Paz, can you help us understand how meta-analyses may help answer this question in the future?
Yeah, so just to echo a lot of what Zoe just said, the meta-analysis might help us better understand what is an appropriate cutoff or an appropriate metric to look at ctDNA. We saw with the different modules in CT Monitor, in the white paper, we were focused on advanced non-small cell lung cancer and immunotherapy-based regimens. And we saw in some of the CT Monitor modules that reduction in ctDNA could be associated at a patient level with long-term outcomes. But we also saw with tyrosine kinase inhibitors that maybe clearance of ctDNA might be a better measure of looking at ctDNA. So a meta-analysis might help us better understand the appropriate metric to be using for a particular disease setting and therapy.
Sure. Can I bring up a point?
Yeah,
just
absolutely.
coming back to one of the things we were talking about before. We heard Anand talk in detail about some of the assay characteristics and validation requirements, and then we're having a good discussion now about what is the unit of metric, right? And how do we measure what is significant? 50%, 90% reduction, complete clearance. One of the things that we really want to emphasize here is that 50% reduction looks very different if you're starting with 20% tumor fraction versus whether you're starting with 2% tumor fraction, right? And so considering the assay characteristics and the precision and reliability of that measurement is actually pretty fundamental to be able to answer that, right, with appropriate rigor. And there's wobble in any assay, right? There's imprecision in an assay, but being able to understand that is really important. And we can measure analytic variation by setting up analytic validation studies. There's also biological imprecision, however, as well. And that's probably something that will be uncovered through future research, is how reliable is this metric, whether you drew blood in the morning, versus later in the afternoon, maybe after the patient just ran on a treadmill perhaps, right? So different shed rates could impact this. Those are things we don't yet quite understand.
Absolutely, and it's not just different shed rates, it's also the assays and the sensitivity. And we had talked about how it may not just be absolute change, but it may be relative change that may be important in the future. I think 50% and 2% example is so relevant and key to that discussion. Carol, we're asking a lot of our patients, right? I think with every clinical trial, we're asking for biopsies, and in some ways, collecting blood almost seems easy. But what are your perspectives, and what can we do to help inform patients in a little better way about what we're trying to do?
I think there's a great deal of interest in the patient and the advocate community about ctDNA for those reasons. It's not invasive, it's also not an imaging, there's no radiation, any of those things. However, I think patients in these trials will need to understand what a meta-analysis is, the fact how their data will be used, and also that they may never know what their own ctDNA levels were to begin with, or were on treatment or not, that is not the point of this study. The point is really to have data that can be part of a trial that is then used in a meta-analysis, which I used to teach research methods to teachers, and I would tell them a meta-analysis is a study of studies. And so we're going to have a big study based on other studies. So I just think that as patients give their informed consent, it needs to be very clear to them what is being asked and what the outcome will be. And I think people will be very likely to want to enroll and stay with it, because I think everyone involved in the cancer community always wants to give back, and this is one way to give back for the future for people who are coming after them. So I think it's just important that patients know what they're giving their consent to.
And I think in the same vein, I think we should talk to sponsors about making that clear and making the process transparent in terms of how different samples are going to be allocated for research in the future. I think such an important point. I do want to circle back a little bit about calculation of molecular response. And David, picking up on your sort of suggestion about assessing molecular response and looking at absolute versus relative change, maybe Anand, I'll come to you and tell us what you consider as biologically relevant here.
Oh, okay, so you're asking me.
Yeah.
So I think Dave already touched on a few points I'm going to sort of talk about as well. And a key consideration is determining what values are biologically relevant. As I discussed earlier, at low VAFs, such as values below LOD95, the computation of delta ctDNAs may become unreliable. At low VAFs, as Dave also mentioned, the changes may reflect the variability of the assay, the imprecision of the assay, and not any real biological difference. Also, for low VAFs, there may be a large difference in the delta percent VAF, such as when you go from 0.2% to 0.1%, which is a 50% decrease. However, this is only a small absolute value difference. So these facts may be considered as well when we think about the metrics being used.
And Leon, could you comment on how we handled how different treatment arms may be compared in the future state when we have all of these data?
Sure. So we did not recommend a specific approach to compare the treatment arms, and that is because from the studies conducted to date, a definitive approach has not been determined or has not emerged as a winner. So we are, however, recommending a molecular response as well as a depth of response as potential two options for sponsors to consider in future studies.
That's great. We're actually a little bit ahead of time, but that just means that we'll open it for questions and expect our audience to engage with us. But before we open it for questions, looking ahead, what do you see as the most actionable next step or maybe biggest hurdle in advancing the use of ctDNA as an early endpoint in clinical trials? So Zoe, I'll start with you, and then I'll just go down the line. So Zoe, your word first.
All right. So yeah, in my perspective, I think it is important to try to improve our prediction of overall survival and other survival-based endpoints with our liquid biopsy technologies, but the hurdle is that we don't know what the best method will be. It could be, novel or more sensitive assays. It could be different or more time points. It could be different analysis approaches or cutoffs. And we really just don't have the data yet to say how to improve that prediction. Saying that, that's what keeps the field really interesting. And I think that the biggest step here is for us to harmonize around assay reportables, and not just per sample reportables, but the assay characteristics as well as around time points, because we can't go back and rerun our clinical trial and collect different time points. So I think this is the most important step right now, is harmonizing, and I also think this will end up being a useful model for other settings where ctDNA also shows promise as an early endpoint. Thanks.
That's great, Zoe. Dave, we'll come to you.
Yeah, I mean, for me, I think just recognizing what we're trying to do here, it's really about accelerating the right medicines for the right patients and using this as a surrogate endpoint to speed up that process, right? And as we see sponsors move their clinical trials into early stage disease and patients living longer, it's going to emphasize the need to have these type of endpoints at an earlier stage so that they associate with those clinical endpoints. As my good friend Jeff Oxner dutifully said this morning, let's not wait on interim data. Let's figure out a way to use this information to actually make the right decisions based on these interim readouts, right? These surrogate endpoints. So how we connect the changes in ctDNA levels with the right clinical endpoints, whether it's progression-free survival, durability of response, overall survival, just making sure that we're doing that in the right way and actually accelerating paths to approval for the right medicines, I think, is the most important thing to me.
That's great, Dave. Leon?
Yeah, for me, the most actionable step is the low-hanging fruit, and that is just the continued refinement and harmonization of ctDNA assays that is consistent with our recommendation that was put forth by BlockPack.
Anand?
Yeah, so in terms of overcoming hurdles, I think that the development of reference materials to help harmonize across disparate assays may be helpful in overcoming the challenge of pooling ctDNA data across a wide range of assays. Another actionable step may be defining the performance characteristics, for example, the minimal LOD95 levels for tests that detect ctDNA changes.
Carol?
Well, I agree with what Dave said, that this is a very exciting project in terms of being able, the overall goal of being able to use this as an intermediate endpoint really would accelerate, effective treatments for patients. And I think that's so exciting. I think in terms of one actionable step coming from the patient's point of view or the advocates, I think is really to make it clear what these trials are about, to help recruit people into them, and then provide support so that people are not dropping out of the trial, so that we can have robust data reporting and hopefully good outcomes from it.
So important to get everyone involved. Absolutely. Clear communication. Paz?
Yeah, so one potential hurdle that I think we need to continue to think about is particularly in the metastatic setting, again, how are ctDNA endpoints going to potentially provide an advantage over imaging assessments or endpoints related to imaging assessments, or can it potentially enhance endpoints that are based on imaging? Can it be used instead of imaging assessments? So these are things we're going to have to carefully think about. And then next steps, I think what we're doing right now is one of the most important next steps, just talking about the data that we need to generate moving forward so that we can pool data to better understand the relationship between ctDNA and long-term clinical outcomes.
All right. Very good final thoughts, but we'll open it up to the audience.
Can I add one more thing?
Oh, Carol, sorry.
I just, this is only my second time at this meeting, and today, hearing the first two panels, I thought, so much about clinical trials that we can fold possibly right into this in terms of the decentralization, the pragmatic aspects and so on. I think that those are all, I said to someone, I can see where our panel was going last because all that came first and it was so important.
It's great. All right, this is going to be a very interactive Q&A session.
Thanks. I'm Paul Klutz from the FDA, and I have a question. Objective response rate, one of the hugest benefits of objective response rate is that causality is not an issue in single-arm cohorts. And it's because we all assume that tumors inexorably grow if not intervened upon. Is there data on ctDNA regarding sort of whether it waxes and wanes more than, say, tumor response with respect to differential shedding, or if you may get some reduction in ctDNA for reasons other than therapy? And I know that's a hard question. You'd have to sort of serially, test people who aren't being actively treated, but that's an important point as far as a single arm use in a single arm as a single arm metric. So my first question is about causality, and Paz kind of got to my other point, which was if the T2 data is the strongest data and it's 7 to 13 weeks, and we get our first imaging assessment at 8, at least in the metastatic setting, it's hard to see the time savings, especially when we consider that duration of response is really important too. So we end up wanting to see that it's held for months thereafter. So I guess first question is, do you guys have much on whether it waxes and wanes over serial testing of untreated patients? And second, how you might tackle this tension between really early information, but then it's not really important unless we can keep that level down over time.
So maybe I'll take a first stab and then I'll open it up to the panel. I think we don't have a lot of data on serial testing in untreated patients, but we do know that the dynamics can vary based on the treatment type. So the immunotherapy treated patients may have different ctDNA dynamics compared to your chemoimmunotherapy patients, and then TKI dynamics may be very different. But it's a very good question. I don't believe this has been looked at in untreated patients. And maybe, Paz, I'll ask you for the second part of that question about the timing.
So the timing. So again, in this white paper, we had talked about doing a baseline assessment of ctDNA and then 3 additional early time points on treatments, and then also an assessment at 6 months. And there is some thought that looking at ctDNA at 6 months might also be correlated with long-term outcomes, that it also might be a way to measure durability of response, but this is something that at the FDA we are really interested to learn more about, and something that a meta-analysis could help us gain more understanding of.
Anyone else from the panel? Zoe?
Yeah, I could chime in from the perspective of industry. We've seen some of these ctDNA technologies can precede radiographic relapse by 3 months, 6 months, or longer. And so I think, a lot of this conversation's around setting ourselves up for meta-analysis to establish surrogacy, but something else that on the drug development side we're really excited about is to have another tool to look at efficacy in early phase studies to help with this go, no-go decision making around if you actually have a molecule. And so I think as long as there's some signal there that ctDNA can predate the imaging result, that it does seem to hold promise for at least that drug development angle.
That's great. Diana?
Hi, everybody. Oh, I moved this. Just for Paul's question before, we published...
Diana, do you mind introducing yourself,
yes.
Name and affiliation?
Diana Marino-Vega from AstraZeneca. And there was a small study on looking at pre-treatment variability, background variability in ctDNA. So there's a little bit, and obviously it happens more at LOD levels. So that will be published soon. But I think that makes a great point in that we need to be able to understand it fully. We need to be able to understand how ctDNA works fully, but we're getting there. We're building all those cohorts. We're building all the data set. I'm just very happy to be here today because, full disclosure, I was part of the white panel discussion and in previous CT monitor work. But I think that it's really exciting, the point that we're at now, that we have clarity from the FDA of what they're looking for. We're putting together those frameworks of knowing how to collect the data so that we will be able to meet the data requirements that the FDA is requiring. But as we are challenging the sponsors to incorporate this framework of data collection and plasma collection into these trials, these prospectively collected trials. Because at least that's the way that we're seeing it. Like, this needs to happen at the sponsor level in order to be able to put all this data together. Could you comment a little bit about the common elements that were derived from an advanced stage setting into future trials that are potentially looking at earlier stage setting, which is very important because that's where we want to go. We want to go at a place where we can cure patients, and that seems to be more and more in the earlier stage of disease.
Yeah, I'm going to just piggyback the virtual question also here, because as you point out, I think there's a great potential to use this in the neoadjuvant and adjuvant setting, and would love to hear from the panel how we would apply these findings to early stage. So David?
Yeah, I'm happy to take a crack at this, Diana. I mean, to me, I think a lot of the framework that we are putting in place for late stage disease can probably be directly applied in early stage disease. There's going to be some key differences. Assay sensitivity is a big difference there, right? And so when we think about percent change and what is meaningful, again, going from 20% tumor fraction down to 10% tumor fraction, the assay doesn't need to be that sensitive in late stage disease where shed is much higher. In early stage disease, where we're talking about an order of magnitude at least less shed, we need to have more sensitive assays, right? And so bringing the assay sensitivity way down to make sure we're covering that is going to be a key difference, obviously. But again, the percent change, the overall change, those things may still apply, right? The framework of timing of collection, those things may still apply. So it's nice to have a framework here that we can think about applying in early stage disease, but understanding that the technology itself will certainly evolve.
And Chara, I just want to follow up a little bit too. In May of 2022, the FDA did issue a draft guidance looking at ctDNA in the early stage of solid tumors. Again, thinking about the potential regulatory use of ctDNA. In the metastatic setting, we have things like overall response rates that we can use for accelerated approval. We don't have something similar in the in the early stage setting where we don't have anything on imaging that we're following oftentimes. So that is why I think our draft guidance had been focused more so in the early stage setting. So again, not to say that ctDNA endpoints can't be developed in the metastatic setting. I think there are potential uses there, particularly if you don't have any measurable disease, but did also just want to point out FDA's kind of thoughts on this as well.
Yeah, and I agree, David. I think the framework will still be the same, but the technology is moving so fast. We're already seeing data being read out in the early stage setting. So potentially a CT monitor type approach to try and see where we are with the aggregate data so far, and then defining it might be a next key step. But I think this is a great question. We'll move on next to the microphone on the right.
Hi, thank you. Hi, my name is Kelly Oliner. I'm from AstraZeneca. I was really struck by your use of the term absolute change versus relevant change. And I do understand measuring around the LOD and what kind of problems you can have there. But where I was hoping it might go is that there might be some tumors where looking at specific changes is a lot more relevant than just looking at VAFs. And I'm kind of wondering, if, for example, like EGFR-driven tumors, right, looking for resistance changes, those sorts of things. Is there a plan to be kind of looking at that more specifically, or are you really focused on kind of the meta-analysis of any ctDNA?
It's a great question, Kelly. Maybe Anand, I'll come to you on absolute versus relative.
Well, Kelly, I think you were getting at more specifics. Can you rephrase your question?
I was really struck by the term of absolute change versus relevant change. And I thought the panel was going to start talking about maybe specific changes instead of just VAFs. So I was wondering if that's been discussed or not.
So the panel was talking about absolute change versus relative change, not relevant. So relative being the delta ctDNA, right? And I think I discussed some of the issues around the delta metric, right? And as far as I know, we're not looking for changes in any driver mutations or resistance mutations in this particular project up to now. But that's something you may be exploring.
It's an important consideration, though. I'll say, if you're looking at a lung cancer that's driven by EGFR, is it more important to track the clonal EGFR mutation than it is to track the variety of subclonal variants, right? And this gets into the balance of like, how much do we want to sort of constrain the technologies by defining the metric that they actually use versus allowing adoption of different ways of calculating whether you're taking just a median allele frequency approach or you're using a more sophisticated algorithm that incorporates fragmentomics and methylation and genomic alterations to compute a ctDNA sort of fraction value. So it was that balance of, how much do we not try to constrain the technology versus just talking about, the metric as at a higher level.
Thank you, Kelly. We'll go to Jeff.
Jeff Oxnard, Eli Lilly. I'm sure many of us in development in the audience are doing this now for our trials, right? We have these data, we're excited about the ctDNA changes early in drug development, and it makes us think, wait, maybe this drug's a breakthrough drug, and maybe this drug's got the right dose. We're using validated metrics to help us understand our drugs. Does the FDA want to see these data? Like, if we package them up and put them into a briefing document, would the FDA be like, wow, we really appreciate seeing all this extra nerdy data? Or would it be like, this is a total headache, this stuff's not validated, please don't put this in front of us? You know what I mean? Like, are we, because you guys have appropriately said, this isn't quite validated, so should we not share the data? Or would you rather us share the data and then you can reply in your response, actually, this is not validated, you shouldn't have sent it? Like, what do you think?
So Jeff is really asking, should he package some data and share with you, Paz?
Absolutely, we would love to see the data. I mean, obviously in a way that we can digest and make sense of, but we have seen things before like, this is the data that we have when someone has a ctDNA positive result at 3 weeks versus 9 weeks, and this is our internal data, and how do we think about this, even when you think about using ctDNA as a stratification factor, so we want to see this data in your thought process. We do want to see the data. Yeah.
Including for dose selection? Could a dose selection submission include this kind of data to complement?
I think that warrants further discussion, but definitely could be something that we'd be interested to talk about.
All of this is more proximal applications, earlier than surrogacy, but there are ways to initiate some of this sponsor FDA dialogue. Maybe.
Right, and for sure, there's a difference between using ctDNA as an endpoint for regulatory purposes versus enrolling patients based on ctDNA status. We already are seeing trials where patients are being enrolled because they're ctDNA positive. So that's already happening. We're okay with that. If there's, again, a rationale for that. So these are things that we already are discussing. There's different uses of ctDNA, and we'd be happy to talk to sponsors about it.
Part of the totality of data, right?
Yes.
It's sort of an easier label. Okay, thank you guys.
That's great. We have about 2 to 3 minutes left. So I'll ask all three of you, you've been very patient, but let's have these questions be really brief. So go ahead, microphone on the right.
Thank you. Arunava Chakravarti from Novartis. As I was hearing this discussion, some of the terms that came up like molecular response, the choice of absolute versus relative change, I was reminded like, while this is new, but it's also not very new, because if you go back to the times when imaging endpoints were defined, like based on RECIST, like RECIST itself is based on some very quantitative threshold based on what you define a responder or a disease progression, or whether it's a target lesion or non-target lesion. So my question to you guys is that as you guys are developing this framework we're at an early stage, how are some of the methodological principles that were built in, like RECIST 1.1, 1.0, like from bidimensional to unidimensional journey, how is that playing out in the CT monitoring project? Just love to hear your thoughts.
It's great. So that's an analogy that we actually used during one of our initial discussions for this working group of how we can eventually use ctDNA as another test, right? Could it be used as we use a CAT scan or a bone scan or a PET scan? And some of the principles were really aligned with lining up the time points so that we can get alignment. And really, I think the next step will be how do you define the molecular response? Because in RECIST, it's very clear. There's 5 lesions. They have to be a certain millimeter. They have to be followed. They have to be defined at baseline. So I think there are a lot of parallels that were discussed. I think that's a great point. I'm sorry, we'll have to move on to the microphone on the left.
Hi, I'll be quick. Callie Gordon from Boudica. I have a question about the baseline sample. So anecdotally, I've seen where prior treatments, and if there's not enough of a time period before you take that C1D1 baseline sample, it might be negative. do you have any recommendations around the timing between, let's say, a prior therapy and the new trial? And do you have any sort of research studies focused on that from that perspective?
Zoe, I'll go to you.
I think we do have that in the paper, but I can't recall right now what the timing is. Yes, but we do say that it's best to collect that baseline sample within 14 days of the treatment initiation. But your question of how much before that can you have a prior therapy, I think we cover that, but I'm afraid I can't recall the number right off the top of my head. I don't know if anyone else remembers.
I do think we said 14 days, but we'll double check for you.
Between the prior therapy and the new trial?
Well, these were treatment naive patients, so we were saying prior to initiation of therapy.
Okay,
Yeah.
that's helpful. Thank you.
Thank you. And last question.
Not a question, just a comment. So we didn't necessarily say when treatment could have been from the last one, just that it should be reported. We didn't necessarily go down. We know that there are potential differences in ctDNA measurement after somebody's been exposed to therapy versus someone who hasn't. So it's something to measure and report when it's happening, but we didn't necessarily say a number.
Thank you, Hillary.
Thanks, Hillary. And last question.
Yes, Chris Karlovich from Frederick National Lab. Really enjoyed the discussion, great comments. So I have really more of a pragmatic comment / question. I work very closely with the NCI. We support a lot of their clinical studies, NCI Match, NCI Combo Match, ETCTN studies. We have our own targeted panel. We've tested thousands of samples from NCI trials, and the issue is around cost. If you are going to be testing a baseline sample, C2, C3, C4, 6 months, and then you always want to get the progression one. Don't forget about that, because you want to look at mechanisms of response and resistance. That's 6 tests. And we've been given direction that we ought to try to limit it to 3, maybe at the most. That's what we typically do for most of our studies. I would imagine the cost is also going to be a consideration for other folks, pharma companies. I just wonder if you had any comments about that.
Yeah, I think you got the last word with cost. It's always about cost, right? I wonder if any of our colleagues have a comment. I feel like this is something that we need to collaborate on so that we can collectively bring costs down. Dave, I don't know if you have a
Yeah,
comment.
I mean, and at this early stage, before there's an approval of anything, cost is a clear consideration for how we understand this in trial design. But we have to sort of start with the patient-focused requirement first and understand, let's do as much as we can to collect the information and then maybe whittle down to what is a practical application in a post-approval setting, and then hopefully have a defined reimbursement strategy that enables the cost of that assay to be used in the right approaches.
Thank you so much to our panel, to Hillary for leading us, and the working group members that put together this fantastic white paper as well as recommendations. Thank you, everyone.
Thank you, and thanks to all the panel discussants today. Very briefly, I know we are at the end of our time this afternoon, but before we close out the conference, I do want to explicitly say thank you to everyone, both in the room and online, for joining us today. This working meeting has been really critical to informing and advancing our work as an organization and our community more broadly as we collectively aim to catalyze the future of cancer research and care. Please note a few upcoming events that I think should be on the screen as we move into the next year. And I want to thank everyone who made today possible, particularly the amazing group of people at Friends of Cancer Research for your leadership and working with our expert panels over the last several months who made everything around the meeting today also happen and come to reality. Special thanks to all of our speakers today and to all the working group members that participated in the discussions over the last couple of weeks in order to formulate the white papers distributed for today's meetings. These were meant to be a discussion exercise that hopefully can be informative as these complicated topics are considered in components of drug development into the future. We really appreciate all of your thoughts and discussion today, and thank you for doing so in a very action-oriented way. There's certainly much to do as we move forward into an action-packed 2025 with multiple different data readouts and conferences that we have planned, and we certainly hope that you'll join us for those, and appreciate you being here today. Thank you, safe travels, and have a good rest of the week.